Madam, can you start the session? Satyatruna.
Yes, sir, we can start.
Your ma'am.
Dear participants, very good morning and welcome to the day 2 of International E-Conference on COVID-19 era need and challenges and in healthcare and pharma sector. Today we are having 3 technical sessions. For session 1, we are having Dr. Prajakta Kulkarni, scientist and entrepreneur, and topic of this session will be COVID-19 diagnostic challenges and innovation. For session 2, we are having eminent speaker Debashish Nayak sir. Topic for this session will be animal models for SARS-CoV-2 pathogenesis and transmission. For session 3, we are having Dr. Rahul Kodgule. Topic for this session will be efficacy and safety for COVID-19 vaccines. Now, I would like to introduce our today's first speaker. It would be a great pleasure to introduce Dr. Prajakta Kulkarni. Dr. Prajakta Kulkarni is a scientist and entrepreneur. She has pursued her PhD from North Dakota State University, Fargo, United States of America. She is currently working as Director and Chief Scientific Officer at Thyron Technologies, California, USA. She was postdoctoral scholar at University of California, Santa Barbara, California, USA. She has many research interests, including nanomedicines, bioresponsive material devices for early detection of diseases, formulation and development, translation medicines. Madam has excellent academic record and she has received various prestigious awards and scholarships. So one of that is first place she has achieved as People's Choice Award winner for NDSU Innovation and Challenges in 2016 for designing 3D printed perfusion bioreactor for the personalized cancer treatment. She has filed 4 patents, out of that 1 has been granted as a US patent. Madam has excellent publication, excellent academic publications in very reputed journals and books. She has presented her work in various international conferences. Now I request Dr. Prajakta Madam, please proceed with your session. Over to Prajakta Madam.
Thank you, Madam, for a great introduction. Hello, everyone. Good morning, and thank you very much for coming for the session on a Saturday morning. I'll go ahead and share my screen now. Let me know if you have any problems seeing it. I think it's It's visible now. All right. So today I'm going to talk about COVID-19 diagnostics, challenges, and innovations. So before going into very scientific details, let's have a look at the year 2020. I know everyone, what everyone is thinking is, oh, year 2020, a year of COVID-19. Obviously, yes. It was a year of challenges, definitely. But somehow all of us learned to adapt. And we came with a lot many innovations this year, especially in healthcare sector as compared to any other year. Unfortunately, the number of cases are still increasing. We are still not out of the COVID-19 era. Till today, 175 cases have been recorded all over the world, and we have already lost 3.7 million people to COVID. But let's take a look at how everything started. Remember that day, 31st December 2019, when all of us welcomed 2020. And at the same time, on the exact same day, WHO picked up a media statement about viral pneumonia that was observed in Wuhan. The term viral pneumonia was very scary for all of us. And the reason being that we were not equipped with handling something like this. So we witnessed that there was shortage of ventilators, there was shortage of facilities, and we were really not prepared to handle a viral pneumonia like COVID-19. On March 11th, 2020, WHO declared pandemic, considering the severe severity of and spread and inactivity towards developing treatments for COVID-19. And since then, the fight with this invisible enemy, the novel coronavirus, began. But the virus was extremely novel, right? Was it extremely novel? Technically not, and we'll come back to it later. But it was a novel coronavirus, so we didn't know how it behaves, right? And how do you fight with something that is so unknown? So we definitely need vaccines. All of us knew that. But developing vaccines was going to take a really long time. Till then, what do we do? So obviously, we have to manage symptoms for some time. We have to do some research during this period and figure out what can work and what can't. We have to come up with rapid diagnostics, and many attempts were done to repurpose drugs so that we can manage symptoms. But most importantly, we knew that we have to limit the spread of the virus. And then the entire stay at home, stay safe era started, and we are still conducting all the conferences like this. All of us jumped on Zoom, started meeting each other, friends, families, and professional peers. Everyone was online. But how long can we keep doing this? How long can we meet each other online? How long can we stay away from our loved ones? And most importantly, how long can we stay at home and keep trying different recipes? So governments knew that people have this need to get out, interact with other humans, but still they want to control the spread of the virus. So I'm not going to go into guidelines. All of us are following those guidelines, which is great. So every government published some guidelines on how to behave in this period, and all of us adapted. But there was this great need to go back to normal, and there is still this need to go back to normal. And when there is need, there is innovation, because innovation stems from a need. And since then, the race in the development of diagnostics and vaccines started. This is a timeline. I'll just go over it quickly. So on March 11, 2020, WHO declared COVID-19 as a pandemic. At that time, there were only 100,000 cases of COVID-19. but 114 countries were affected by it. And so looking at the spread of the virus, it was a pandemic. In mid-March, we realized that it can undergo mutations and that can increase transmissibility of the virus. But after that, we started approving, we got many vaccines approved and many vaccination drives started. But during this period, there were a few key learnings that we learned during this period. So first of all, this virus has a long incubation period. So all of us know it by now. It can take 2 to 14 days to show symptoms after exposure. And there are many asymptomatic carriers. So I want to give you this example. So let's say on day 1, someone got exposed to like SARS-CoV-2, the virus, and then got tested on day 3. The test results arrived on day 7. The test was negative. So kept doing all the chores as the person was doing before. On day 10, the symptoms began and then got tested again on day 11 and then was tested positive. So this is a very drastic case, to be very honest. Like most of the patients do start showing symptoms by day 5. But in this case, this patient didn't show any symptoms till day 10 and got to know about it on day 14th. And so for those 14 days, whoever he met, they were also exposed to the same virus. So we knew that there's this 14-day quarantine period and we have to follow the regulations and all those things. But we learned that diagnosis, screening, and contact tracing all are crucial. Another thing that we learned about this virus is how it behaves. So this is a very interesting graph. If you look at this line, the blue solid line, it is showing the viral load of the virus. And this dotted line is showing the immune response of the patient towards the virus. So, and this is a 0. The 0 indicates the time since the onset of symptoms. So for first 5 days, when the patient was asymptomatic, you can see the viral load is increasing. When the patient is showing the moderate or mild or moderate symptoms, that's when the viral load is the highest. And then viral load goes down and the illness starts getting severe or even critical. So when the patient is hospitalized, the viral load is on decreasing trend. And so what we learned is the severity of the illness is related to the immune response that is generated towards the virus. Of course, there is a correlation between the viral load and the severity of the disease or the immune response as well. And that leads to organ damage and admissions to intensive care. And of course, in some cases, deaths. So when we talk about COVID-19 diagnostics, we are not just talking about detection of SARS-CoV-2. We are talking about symptom detection, SARS-CoV-2 detection, biomarkers, which are used here for measuring disease progression, disease monitoring, and variant identification. So all these parameters come under diagnostics for COVID-19. Some of those are lab tests, while others can be point of care tests. So symptom check, I think I'm not going to go over it for an entire year. I think newspapers have done a fantastic job. All of us have used contactless thermometers and oximeters. So I'll skip this one. So coming to SARS-CoV-2 detection, there are different methods. And these methods are based on what exactly are we detecting. So virus has a very simple structure, actually, but and there are only 2 tests which come under diagnosis. So one is the molecular molecular diagnostics. One test protocol is molecular diagnostics, and the other one would be the antigen testing. So if you want to broadly classify this test, these are only the two classes. But then there are many different tests that come under these sections. So looking at the structure, I know yesterday's speaker also showed the structure of the virus, but I'm just going to focus on what exactly are we detecting in this test. So for molecular diagnostics, the area of interest is this RNA. Most of us know this. And for antigen testing is the spike protein that is covering the virus. So molecular tests or nucleic acid detection tests are based on RNA detection. Many people have heard about antibody test, but it doesn't give us information about whether this person has COVID right now or the person doesn't have COVID. What it tells you is, did this person have COVID in the past? So antibody test is basically, so as if you remember the graph, the antibodies start increasing when the severity of the disease increases. And the antibody test is usually seen, is carried out when we are looking for plasma donor, or if we want to find the asymptomatic person. But the rapid testing, which is very famous, comes under the antigen testing. Now, these tests are also based on which sample we are using. So, or basically sample collection is also based on the test we are carrying out. So for viral DNA, we usually go for nasopharyngeal or oropharyngeal swab, but for antibodies, we go for blood sample. So factors we have to consider while selecting different test protocols is, first is the severity of symptoms. At what stage is the patient? Then time, when basically on that scale, when we are doing the test for the patient, and the site of sample collection. And the reason behind site of sample collection is that every site shows different viral load from the same patient. Most commonly, the swab is collected from nasopharyngeal area or oropharyngeal area. Now coming to molecular diagnostic tests. So these are mostly nucleic acid amplification tests. And most common is RT-PCR. Then NASBA, LAMP, CRISPR-COVID, LFA, Sherlock, CRISPR-Cas129 are also used. So I'm not going to go over the entire RT-PCR protocol, but I will give some glimpse of what exactly happens once the sample is given to a path lab. So a path lab will extract the RNA, will reverse transcribe it to DNA, and then they will make it ready basically for amplification for it. They'll add the primer to it. Then the DNA would be built with the primers in it. And then multiple copies would be made along with a fluorescent dye. Then this will be amplified. After every cycle, the fluorescence will go on increasing, and it will be the cycles, like at least 40 cycles in this case, would be done to make sure if the patient has COVID or doesn't have COVID. So when the detection is possible, that's what the CT number shows on the report. So this is how usually the RT-PCR is done. The first protocol for diagnosis of COVID was published on January 23, 2020. But RT-PCR still remains the gold standard for detection of SARS-CoV-2. It is time consuming, but it targets viral genome. There are two different methods by which you can do RT-PCR, but most commonly one-step RT-PCR is done. But it requires a sophisticated instrument. It requires technical expertise, It is extremely time consuming and is expensive. The cycles, which are denaturing, annealing, and elongation of DNA, so the steps in the protocol, those are at different temperature levels. And so there is a lot of time spent in the entire protocol. So the test protocol can take up to 4 hours, but a patient can get their results in 3 days. And so something easier, faster is definitely required. So NASBA is nucleic acid sequence-based amplification. It, it works at the exact same temperature in the isothermal conditions. So there are no heating and cooling cycles involved. So it saves some time. It requires only 2 steps, denaturation and polymerase dependent amplification. And so it's time efficient. The sensitivity and specificity of this method is similar to RT-PCR. And so it's a good test to use. But most popular has been LAMP, loop-mediated isothermal amplification. It is less expensive. It is far faster than RT-PCR, and selective amplification of target nucleic acids is possible. It can be modified to colorimetry. So most of the other methods require like fluorescence spectrophotometer, but this one can be done with the colorimetry, and so it has been a bit more popular. But we still need to innovate, and the reason is there's we have to use these techniques on broad population. We want to screen the entire community, right? So for that, we cannot keep using a test that takes 3 or 4 hours or even like a couple of hours. Also, the cost is a very important factor. Then time required obviously is important. And there are many asymptomatic cases. And so we want something, something that can screen the patient rather than diagnose the patient, or at the same time diagnose the patient. So many point of care devices have been developed recently, which are designed for bedside testing. They are accurate, like that's the necessity. Some are accurate, and the requirement is the lab quality results. They're portable, so they can be easily transported where and when needed. They are affordable, and so you can use those devices on multiple patients. And most importantly, they are next to your bed, right? So you can take those tests at home. So the normal protocol here would be a patient sees some symptoms, collects a swab, transports the sample, or patient goes to the pathology lab, and that's where the swab is collected. Then sample is processed in the lab and the results are communicated. And then the patient communicates those results to a doctor, and then doctor advises this patient to quarantine, and the patient receives the treatment. This is a time consuming process, but instead, if the patient has control over the test, then the patient can take the test and start the treatment immediately. And so LFA, I'm sure most of us have already used something that comes under LFA. So LFA is lateral flow assay. These are simple devices. They can detect the presence or absence of the target analyte in liquid samples. So they are not really quantitative, but more like qualitative or semi-quantitative. So they can detect the presence or absence, but in this case, they work just fine because we just want to know yes or no. They can be used as home testing devices, point of care devices, and also they can be used in the laboratory. The structure is extreme, or like assembly is very simple. They have one sample pad, that's where the sample is added. Then they have a conjugate release or a conjugate pad on which label tag combined with biorecognition elements are added. I'm not going to go into what those elements are. We'll see those later. And then there is this reaction membrane, which has the test line. And that's where all the reactants are loaded. And that's where the actual reaction takes place. And the control line where um, so there is target antigen antibody interaction, which is already taken place, is already there. So once the sample travels there, it is going to give us a control line. And then the absorbent pad, which acts as a reserve waste. So the simplest example for this is basically a pregnancy test. That also is a lateral flow assay. So it exactly works in the same way. And those kind of assays are very easy to work with. But how did we use those for rapid antigen testing? So we tried to use this for antigen and spike protein interaction, antigen and antibody interaction, where antigen was spike protein and antibody was for spike protein. And the results were obtained in few minutes, like in 5 minutes. And so that was called a rapid diagnostic test. It was very useful for screening the large population or even for testing many patients at the same time. It allowed more tests in short time. There were reports on saliva samples being as effective as naropharyngeal swabs, so more convenient. They were cost effective, and the technology penetration was very visible. They were, of course, point of care, but sensitivity specificity was a bit of a question, mainly because of false positive, false negative kind of results. So this is a very interesting hybrid approach where this is a very hybrid approach where CRISPR, which is um, a nucleic acid based test, and which is coupled with LFA. So CRISPR coupled LFA or CRISPR based POC point of care devices were developed. And there were 3 different interesting products that would come to the market. I think Sherlock got the EUA already, but Sherlock, Detector, and Inspector were 3 hybrid technologies that were developed during this period. And they had precision of a nucleic acid amplification test, but at the same time, ease of LFA. And so they can be done in or completed, these tests can be completed within an hour. There were many lab on chip devices that were developed. Advantages of microfluidic devices. So lab on chip devices are mostly microfluidic devices. And if you see this is a microfluidic chip or a lab on a chip, and it requires a very small amount of sample. It flows through these microchannels, reacts with different reactants, goes to the sensor. Sensor detects the analytes or analyte interaction, and then the data is basically transferred to another unit or another electronic platform. This can be converted into high throughput studies. And they can use different sensors like optical, electrochemical, electrical sensors. They're very cost effective. And of course, they can be used for point of care diagnostics. They're very robust and predictable. They have low cost, fast reaction times, point of care capabilities, sample to answer automation. So it was like all win-win-win kind of a situation. Some interesting research, I would say, it's not there yet. So some interesting research was done on multiplexed COVID-19 monitoring, where multiple studies were done on the same device and communicated to an app. Wouldn't it be awesome to actually have something out there that which can test you, and you can read the readings with this tiny little probe, and you can transfer your data here, and then it goes to your doctor, and it's all all futuristic, but really cool ideas came during this period. So most of, till date, whatever I was talking about, most of these techniques were based on either colorimetry, fluorimetry, or electrochemical sensing. An interesting way of testing antigen-antibody interaction is also by, by measuring changes in the electrical properties of the reactants. So there is the I'll talk about just one method. There are 3 different methods which have been used. I'll talk about just one method, the field effect transistor or FET-based biosensing. So what happens here is, so this is a patient and the patient gives the sample. The sample is then loaded on a chip or a sensor. It is a FET-based sensor. And on this sensor, the antibodies are already attached to the sensor. So as this virus comes in contact with this device, the antibodies interact with spike proteins and a change in electrical properties is noted. So the electrical signal increases here as the interaction takes place. And based on how much it is increased, we can do a quantitative analysis. And so this is a very interesting method because we can then have multiple tests running on the same platform. The other methods are potential stat based sensor and voltammetry based sensors. Another interesting technology is nanopore technology. And I was very lucky to get a chance to use this technology. And it was extremely fascinating when I first got my hands onto it. So this is a very small, tiny little device. And you can connect it to your laptop or any other computer. You put your sample here. And in the device, there is ionic. So there are multiple nanopores, like nano-sized pores on the membrane. And you wet the membrane with some ionic solution, and then it is kept at a particular current. Then with the motor, basically you start the device, and from this device, a single-stranded RNA is pushed through the nanopore membrane. As every single base enters this pore, this electric current changes. And it changes based on, so this is just an example. So let's say this red dot goes through it, the current dips once, holds it there, and then goes back again. Then for a bigger molecule, it dips even further and goes back again. So here, if you see that this is a motor, there's a membrane, there's a run condition as in basically ionic solutions, and this is a nanopore reader. So this is where actually the current is measured. And based on those that current, basically, we can figure out what is the sequence of an RNA. So this is a very cool technology, and this can give us results within an hour. And it is like a little thing that can sit on your workbench and do all the tests. Now coming to the antibody tests. So antibody tests are, as I said, it is not a test for diagnosis. It is more like for figuring out the asymptomatic individuals or figuring out plasma donors or something like that. Or it doesn't tell you if you are COVID positive right now, but it will definitely tell you if you were COVID positive before. And it is also based on LFA, so I won't go into details again. Another diagnostics that is required is organ monitoring. So COVID has effect on multiple organs. And of course, we cannot measure all the effects at the same time, but we need monitoring for all of those. We also have to monitor some biomarkers for disease progression. So we looked at detection of SARS-CoV-2. We looked at how to find an asymptomatic individual, but how do we know that how far are we or how far is the patient in that disease progression path? And for that, all these tests are done mostly in the lab. There are some POC tests developed and are more like research articles at this point, but they can be used in the lab settings. And imaging, I think all of us know that imaging is something that all hospitalized patients have to go through to make sure, again, for the disease progression. As as introduced before, so I have founded two companies. One is Chiron Technologies, and in Chiron Technologies, we basically enable smart electronic and digital solutions for personalized healthcare. And the other one is PsyCare, and it is for, so in PsyCare, we are developing point of care diagnostic devices for early detection and real-time monitoring of kidney disease. Both the companies are based in California. And so PsyCare was founded in 2018, and Chiron is slightly new. Both the companies do have certain COVID-related solutions, but due to nature of the meeting today, I cannot disclose the technical details, but feel free to reach me. I would be happy to discuss the innovations that we are doing. So during this era, during this COVID era, total newly approved diagnostic tests for COVID-19 are more than 590. And this is due to our very very interesting regulatory related things. So many countries allowed emergency use authorization for these diagnostic devices. And that's how we got so many diagnostic tests approved in a single year. So challenges going forward. So it's good that all of us are here and we have adapted to the challenges of 2020. And now going forward, as all of us are getting vaccinated, there's still many challenges. So we can look at this stadium, right? And we really want to go, there's an empty stadium and we really want to go to here, right? We want those IPLs to start again. But one single person or one single asymptomatic person is going to turn those events into a super spreader. And so how do we stop that? Or when do we get to go back to normal? When do we go back to go to the stadiums? And what are the things that we should be doing? So there is a difference between, as I mentioned before, there is a difference between screening versus diagnosis. So we can diagnose something when we see symptoms, but we can screen patients. We can screen people who are going to the stadiums. We can do communities testing, and we can enable contact tracing. These all things are just going to help us get into those doors, but not with 100% confidence, right? So diagnosis is more like when there are symptoms, when there's targeted testing, when it initiates with contact tracing most of the times, and the results are mostly reliable. But in screening, the results may require a follow-up testing or follow-up diagnosis. So where are the innovations coming from for the next, like, post-vaccination period? Of course, there would be some innovations again in vaccination. And I think other speaker must have talked about it. I'll talk about screening. There would be definitely some screening related innovations coming up. Then contact tracing, novel ways of tracing contacts, like who did you meet with 10 days back? How do we figure out who did we talk to on the bus, or uh, who did we meet in the last meeting? Then there would be innovations related to variant identification or mutation tracking, which is becoming extremely important. And the most importantly, something that everyone is talking about these days is environmental screening traps. And I think it's a very fascinating idea to have the environmental screening traps. So what does it mean is, for example, everyone is going to a chemist shop, and mostly sick people are going to chemist shop. So what do we know about the viral load that this chemist is exposed to? Or what do we know about the viral load at a bus stop we are going to? Or what do we know about a viral load in a restaurant we are eating in? So these environmental traps or screening traps will check for these viruses in the environment and not on the patients. And I think it's very interesting that it is difficult to ask someone to change their behavior, but it's just easy to put one device somewhere and which will automatically keep measuring the virus load. So I think this is going to be a very interesting solution. And I think it's something that people have been talking about it, but I haven't seen any devices yet. So let's hope something comes up very quickly. With this, I would like to conclude, and I would be happy to take questions. Thank you very much for inviting me for this talk and giving me this opportunity to interact with the audience. And thank you to all organizers who have organized this fantastic event. And I think it's very important to have these conversations as we are reopening after vaccinations. Well, thank you.
First of all, thank you very much, madam, for this wonderful session with full of knowledge. It was very interesting and informative session. Ma'am has covered various vital and novel areas of the COVID testing, including antibody formation, molecular diagnostics, RT-PCR testing, and different protocols of COVID testing, nucleic acid sequence based amplification, lateral flow. Even ma'am, although being from the same field, these concepts are even new for us. So thank you very much for letting us know that how the chip is working in the lab and how it is important. So you have shared a lot of area of the COVID-19 testing, different nanopore technologies. We were visualizing with your presentation that how it is actually working, how organ monitoring is important whenever we are having COVID patient, difference between different challenges and diagnostics in the COVID testing, and in a different innovation in the vaccination. So thank you, ma'am, for focusing uh, to this huge area of COVID-19 vaccination. So here there are few questions from the participants, ma'am. So can we take these questions?
Yeah, sure.
Yeah, there is one question from the participant that what about SARI? Is the death rate more or in SARI or COVID-19? So one of the participants is asking. So in which death rate is more?
Well, I'm not in which death rate is more, as
in SARI
what?
or in the SARI disease or SARI or COVID-19? In
Oh,
which?
I think WHO would have a better idea and better data than I could
Surely, ma'am.
answer this.
One more question we are having, that how we can differentiate COVID-19 from another RNA viruses, because other RNA viruses may also shows positive RT-PCR test. So
Yes,
how we can distinguish?
that is a very good question. And yes, that is a problem with diagnostics. So there could be some contamination. So I think I was reading a paper, this is coming from a paper, So doing the test multiple times and trying to figure out, so multiple times is basically the only answer. So it's not going to give you the right answer in the first test. But unfortunately, what happens is many people get tested positive or negative. So it's not, it's not with the same RNA actually. So let's say 2 different RNAs, it's just, they are not just going to be similar in sequence, right? So every RNA will have, so in the RT-PCR method, basically, we convert it into DNA, cDNA, and then when the DNA, so it will have a primer, which is specific for that particular virus. And so when you do the test, so one time you can have the contamination or something like that, definitely. But when you try to do it multiple times, there might be a possibility, there's a possibility that it could, there's a possibility that there could be something wrong once, but I don't think if you repeat it, it would be wrong. So repetition is a good idea, but again, no one is going to repeat it.
Yes, yes.
Yeah.
Thank you, ma'am. One more question is there from the audience, that whether RT-PCR test is effective or rapid antigen test is effective. So this is
RT-PCR.
okay. And then
So
one more.
rapid antigen test is good for, it is good, but RT-PCR is a gold standard, and mainly because it's more precise.
One more question we have received, ma'am, from Laukush Jadhav, that what is the role of immunosuppressant? Why the other devices we are discussing not used in India? So these devices we have discussed in our presentation are not in use uh, for the India. Then,
Yeah.
so this is the question from the audience.
Yes, there are a few, yeah, there could be different challenges. I think one reason behind that would be cost. Like, so the device, the nanopore device I was showing you, that's something like $1,000 for one device. It's not very expensive if you think about it, but it's at the same time, it might be expensive. So how many are they going to sell in India, and how many are they going to sell in US? That might be the thing. Also, there are regulatory concerns, which we have to think about. For every country, there would be a regulatory concern. So cost, regulatory related pathways, and of course the need. So as I said, there should be a need to do something. So there has to be need to get things done. And I think for population like we have in India, it's easier to do rapid diagnostic test. And when we want like precise results, we want to go for RT-PCR.
Thank
So
you.
it's
Thank you
also the
so much.
population.
Thank you, ma'am, for satisfying us with your wonderful answers. On from the from organizing committee of the Anand College of Pharmacy for international conference. We thank you, ma'am, on behalf of organizing committee of International E-Conference and the Anand College of Pharmacy for delivering such a fluent and very knowledgeable session. Hereafter also we are expecting that you will be there for us and our students. So thank you. Thank you very much. Now I
Sure.
request.
Yeah, thank you.
Thank you, ma'am. I request Dr. Mr. Vadulkar sir to please introduce our next speaker. Over to Vadulkar sir.
Thank you very much, Tiwari ma'am. Very good morning to one and all. I Wadukar RD, Assistant Professor on the behalf of Dayanand Education Societies, Dayanand College of Pharmacy and Dayanand Institute of Pharmacy. Welcome you for the second day of the International E-Conference on COVID-19 era, need and challenges in healthcare and pharma sectors. It's a great pleasure for me to introduce our speaker for this session, which is going to talk to us about the animal models for SARS-CoV-2 pathogenesis and transmissions. This is a subject in which we should all be deeply interested because everyone is panicked about to know the facts and how to prevent this pandemic disease like COVID-19. Our speaker, although having spent almost his entire career in the study and research of virology, immunology, and the development of various biomedical devices. Ladies and gentlemen, please join me in welcoming him. Before starting his inspirational sessions, let me introduce Dr. Debashish Naik. He is currently working as Assistant Professor and HOD of Bioscience and Biomedical Engineering at IIT Indore since 2019. He holds the PhD in Molecular Virology and Viral Pathogenesis from the University of Nebraska, Lincoln, United States of America in 2009, and post-graduation from Chennai in 2001 and graduation from Bhubaneswar, Odisha in 1997. Sir has received 11 prestigious awards and honors to date from Indian and US government, like Fellows Award of Research Excellence from the National Institute of Health Science, USA, American Society for Virology Travel Award from USA, American Association of Immunologists Travel Award from USA, and Chancellor Doctoral Research Fellowship Award from the USA. So these are the many more prestigious awards is honored by our eminent personality today. Sir is the principal and co-principal investigator for the various projects. Near about 8 projects handled by the doctor, and out of this 8 projects, 3 projects is already completed, and all this project is funded by the ISMR, DFP, CSIR, DSP for the development of vaccine and monoclonal antibody for various pandemic diseases, including chikungunya, COVID-19, and SARS. Sir has presented 14 oral research papers in various national and international conferences across the world and published more than 42 research papers in peer-reviewed journals with good impact factors. Sir has also filed 5 patents till date. Out of this, 1 patent in on COVID-2 with the topic of neutralizations antibody and its applications. Sir is recognized as a research guide for PG and the PhD students in IIT Indore. Sir had supervised till 3 students for doctoral degree and 4 in current. Sir is not only excellent in academic and research, but also he is a good administrator, entrepreneur, partner, and working in a collaboration with national and international pharma industry, such as Bharat Biotech International Limited, for then Entas Pharma, Aditi Bioscience, yes, and all these industry, they are just developing the antibodies or the monoclonal antibodies or the vaccine for the development of the SARS or for the development of the COVID-19. Sir is a life member of the various professional associations, like life member of Indian Immunology Society, life member of Indian Veterinary Association, American Society for Virology, and the American Association of the Immunologists. With these brief introductions, I would like to hand over the session towards such an eminent, versatile personality, one and only Dr. Debashish Naik. Sir, session is over to you.
Yeah, thank you. Can you hear me?
Yes, sir.
Okay, thank you, Raghu, and thank you, Yogesh, and most importantly, the organizers, such a wonderful introduction, as well as the opportunity to talk to you and share with you what is going on in the corona and animal related works. So before going that, I'm sharing my screen. So I hope you can see these things, and subsequently, can you see the slides now? Which is
now it is visible, sir. Yes, perfect.
Yeah, thank you, Yogesh, and thank you, Raghunathji, and it's a wonderful introduction. It's my pleasure. to
Yes,
interact with you. So as we see that the speaker introduction is already given nicely by Raghu, and I thank you for him again. And now let's go to the topic of my presentation about the animal models and SARS pathogenesis in transmission and in other studies. So my background is a veterinary science. I'm a veterinarian, and my laboratory focuses on One Health approach to the emerging genetic diseases as well as the animal welfare. So this is my theme, and last 20 years I've been working on this theme. And we do several works related to genetic viruses, such as ORF viruses, SARS-CoV-2, Nipah, Zika viruses, where my PhD students are working. In addition to, we do biomedical device development. Here is one such testing we are doing in the animals. We also participate in a national wildlife conservation program, such as vulture conservation program. For the first time, we make a interspecies between chicken and dog, and subsequently we are using that platform to conserve the vultures, which have been going to be extincted or at the verge of extinction in India and endangered species. So this is briefly our work. And to go in an academic way, this part of the talk or this today's lecture is divided into 4 parts, where we are talking about the relevance of animal models in the context of a viral disease, about the mechanism, how the disease is being reflected in the virus, same as in human compared to the animal model, how the animal is being used for different purposes, such as discovery of drugs or antiviral molecules in this current context, how it is being used for the vaccine development, and what are other avenues, such as looking at the zoonosis, looking at the reservoir, looking at the eradication program. All these aspects we are going to cover very briefly in this particular lecture. So the first part of this talk is about the virus pathogenesis, or we call viral pathogenesis. This is one of my favorite topics, where I did my graduation. And we are telling you that we know the viruses existed before the human arrived in the planet Earth, and they will be there even after the human beings also goes out the Earth. So in nutshell, viruses are the integral part of our existence and ecosystem. For example, if you look at your genome or any animal's genome, 97 to 98% of the genome do not code for any protein. Only 3% of the genome is producing some form of gene or any regulatory elements. 97% so-called, we call junk DNA, is typically arrived from the insertion of the retro elements, the insertion of the viral DNA or RNA to our genome in subsequently during the course of evolution. For example, currently we are facing the pandemic since last 30 years about. So today we are talking about pandemic about corona, but we are facing a pandemic of HIV since last 3 decades. Okay, and we know that the HIV is a retrovirus, and it injects or inserts its DNA or genome to the host, to us, and it stays forever. So like that, if you look after 200 years, maybe 300 years, all of us will have a retroviral element in our DNA, because if a mother and father get having the retroviral DNA in the genome, ultimately the babies will get, and subsequently that retrovirus will become you. And a classic example of this concept is also been postulated, and proof of principle or the concept is done in New Zealand and Australia. There are a particular animal population we call koala. Just that Australia and New Zealand separated by sea. So one location, koala is already having one retrovirus in the entire genome. So you cannot distinguish the virus from the genome. It is there, it is dead, it has mutated, no infection is produced. But in other case, it is just infecting the same way the HIV is infecting the human being. That means in long run, the one after other, the viruses come and go, and we evolutionally remain fit and relevant. So this is a larger context that we should not take that the viruses are always bad. Of course, some viruses in some conditions, but it is the way the human evolution or the animation accompanying various genes from here to there, because virus can take a gene and insert it. They are the elements which is doing the job very precisely and efficiently. So we are the viruses. Our genome contains viruses. They are everywhere. Whether you talk newborn, you talk the animals, you talk birds, fish, anywhere you go, living organism, there are viruses. Top of that, the virus mass is the most abundant biomass in the planet Earth, because if you look at the water, if you take 1 ml of water, you will get nearly 1 billion particles of a virus. depending on which location. So many types of viruses, 400 types of viruses are there. Even we don't know about the existence of another 3,000 viruses. So we are only 10% of our knowledge about the viruses what been available. Coming to our topic of SARS coronaviruses, it is also projected that the bat may be containing somewhere between 150 to 300 types of coronaviruses, and we only know 3 or 4. Okay, so that means our knowledge of understanding on the subject of coronaviruses is also limited. Only it is being surfaced very recently because of the pandemic in the past in 2003, March in 2009, and as well as 2019 COVID. We are now knowing about corona, but it is existed long before, and it will be there. Okay, so this is the context we are talking about the viruses. So any work we need to do, particularly related to pathogenesis, we pay great attention to one of the great Italian Robert Koch, who has postulated that if you want to work on an animal, or you want to work on an organism, microorganism, bacteria, viruses, fungus, parasite, so you have to follow certain principles and postulates or guidelines to prove that you are on the right track. For example, you have to identify the organism, okay, and you have to isolate that organism from the source, and you have to inject or introduce that organism to another host, and take the organism from the new host. So a phase 1 host is the original wild type host where the organism was found. Phase 2 has reintroduced artificially, same organism will produce the same disease. Unless these principles have been adopted, we cannot tell that this is the causative relationship. So for the SARS corona, for example, we have to give more stringency also, because the new method of this Koch postulate, which is talking about viruses, requires 6 steps to prove that SARS-CoV-2 is the cause of COVID-19. So what steps are there? First of all, you have to isolate the virus from the diseased host, whether it's bat, animal, human being, pangolin. You have to, you have to isolate. You must isolate. Second, you have to cultivate that virus in a cells. So whichever host is being susceptible to that virus, suppose you derive the epithelial cell line from me or from a monkey, that virus which is being there in me should be able to replicate. If it is a hepatitic virus, probably it will replicate in the liver cells. If it is a virus for the skin, such as epithelium, like a pox virus, it should grow in the skin cell. So you should be able to cultivate that virus artificially outside the host in certain cells. Then proof of filterability. So if you remember the virology, that the virology started when the Ivanovsky passed liquid containing viruses from tobacco mosaic infected plants through a filter where the bacteria can be kept out, but sub-particles below 1 micron will pass through. So that means you have to prove that whenever you are collecting the viruses or microorganisms, you should pass through a filter where the larger size will be kept out, sub-micron, which is around 100 to 200 nanometer typically, will pass through, and that will be the containing fragment where the virus is. Satisfying these 3 is not enough. Then you have to produce the comparable disease condition in the second host. That means if SARS virus cause respiratory illness within me, cough, fever, illness, problems in the multi-organ, organ failure, coagulation of the blood, you have to identify a similar animal model, maybe a monkey, maybe a ferret, maybe a chimpanzee, to show that the similar condition of the disease is being replicated in the second host. Okay, so that is also one such principle. And after the disease is gone to the same host, you have to find the same virus, original virus, which has been introduced to it. So that means you have to go to the full circle. You get it from the human, send to the monkey, identify and show that monkey also get the same disease. After the monkey gets the disease, you recollect the virus and found that the original virus which was introduced is the same as the virus which is coming out. So you have to go to full circle. In addition to the last stringence is that you have to detect the virus in the immune response to the in a manner. That means your new host or the human being or the coronavirus bat or containing bat or monkey or ferret should have some form of the innate immune response or an adaptive immune response to the virus. It should not be it should not be like a bus that you enter in and you go it out. Rather, it should produce some effect. So that means your host able to recognize that one in terms of innate response such as cytokines, or adaptive response, such as T cells and B cell response, antibody response. So all 6 parameters, when they meet, that new postulates, which has been modified by Rivers in 1934 in USA, who is a modern father of the modern biology. He postulated that only when the 6 criteria met, then we tell that a disease being associated with a particular organism. SARS-CoV-2 is associated with COVID-19 after fulfilling this criteria. Okay, so now let's go to this topic. Okay, why is that SARS so important, and what the way things are going around us? So angiotensin, converting enzyme 2. It's a very famous word. I think it is one of the top 10 Googled words nowadays, called ACE2. Okay, is the sole receptor and primary major receptor which is engages with the virus to take into the inside the host. So as we know that a virus just cannot enter the host because there is a barrier in the cell. The barrier are different types: plasma membrane type, then we have lipid lipopolysaccharide, there are physical things, mucus, all these things. But how can the virus go? Because they identify a protein primarily with a modified protein, maybe glycosylated protein, so that they engage to each other and subsequently they internalize, and the genome of the virus is released, and the cycle of virus replication begins. Subsequently, the more and more viruses produced, and so on and so forth. So here in SARS coronaviruses, the most important element is the presence of ACE2, which makes all of us vulnerable to this virus. Wherever this ACE2 is there, likely the virus will go on. Okay. So that is the essence of any animal model. So you remember that. ACE2 is essential for virus to get in. Without ACE2, we cannot culture the virus in vitro or in cells. If the virus do not go to the cells, we cannot culture it, or we cannot produce the disease in animals if there is no ACE2. So this is the heart of this, any animal model or any laboratory study, that ACE2 should be there. Okay. And also there are studies which have been shown that, like you can see here, the cancer situation, people with lupus, smokers, obesity, diabetic people, they have a higher production of ACE2 in their cell. So more ACE2 receptor are being expressed in the cell, which is enabling them more virus to get in and more production of the disease. So also the severity of the symptom. So we can now correlate that, more ACE2 expressed, higher is the chances of getting COVID, and damage to the host is much more. So this is how we are telling that people, those having associated secondary causes of comorbidity in your pharmacological term, should be very careful in this case of coronavirus. Okay. Then is the ACE2 available only in human being? Where are they? Okay. So there are beautiful papers, and in many papers, and I'm just giving you some of the example. So what the researchers have done it here, that they have collected the nucleotide sequence as well as the amino acid sequences of ACE2 receptor across several species. Okay, so they just aligned. We call it cluster W or sequence alignment. Very simple algorithm. So you can align it, and you can see that topmost we have the human ACE2, and it is showing between 24 to 350. I'm not giving the whole detail, but compared to the human, the next one is your rhesus monkey. Then we have marmosets. Then we have pig, goat, dog, cat, pangolin, mouse, koala, all these things. Okay. So the point is that human receptor is not exactly present in the same manner in other animals. Okay, there are certain kind of mutations. Only closest is your rhesus monkey. So rhesus monkey does not have any change, what being shown here, but every other animal has a certain change. So theoretically, now we can conclude that rhesus monkey could be the best model to study the SARS virus pathogenesis. So this is the basis where we go methodically to choose an animal model. Okay. Then the researchers do this phylogenetic analysis. So phylogenetic analysis means that what is the relatedness of your receptor, my receptor, with a pangolin receptor or monkey receptor or pig receptor or koala receptor. So they construct the tree, and they found that there must be, because we all evolved through the evolution, so there must be some common origin. And once the origin is started, then like a tree grows, the branch after branch after branch. So what is that branch pattern and how closely related? As I mentioned earlier, the lower figure can show you that how the rhesus monkey and human beings are very closely associated. You can compare that, compared to that rhesus monkey, in the topmost is your chicken and duck, and hardly there is any correlation between them because the tree pattern is different. Okay, so this is a nutshell that I'm showing you that how the EC2 receptor is present in the nature. So they are there. So we have to now identify which one is the best. So we also know another thing in the SARS virus is spike protein. That's a buzzword, and there is no need to mention about what is spike protein. But there is one component in the spike protein which is essential. We call it RBD, receptor binding domain. That means that part of the spike protein out of 5,000 nucleotides or 1,700 amino acids, a portion of the protein binds to the receptor. And in that portion of protein, there are certain specific amino acids present at 417 position or 502 position. They are specifically do this crosstalk. Okay, so they specifically do crosstalk with the AC2 receptor. Similar, when you have interaction between two, these are the conversation happening between two amino acids. Okay, so if that conversation is not strong, okay, then the virus cannot attach. If the conversation is strong or interaction is strong, virus will attach. Even single mutations can derail it. Even suppose there are 10 amino acids are interacting very strongly, but one is not interacting, that will not also allow virus to get in. So these three amino acids are like 31 position tyrosine. Okay, if you change tyrosine to lysine, it will not work. 41 position also, 353 position. So these are the scientists have identified that if these are very sensitive positions, if you do a single change to this mutation, then you are drawn. So your interaction will be not so helpful, and you cannot allow the virus to get in. It will attach to some extent, but it will not enter it. Okay. So based on this approach, now we know that this koala cannot be, a New World monkey cannot be, a mouse cannot be. Then how to do the animal study? Well, before going to the animal study, we also to know that what else is required. For example, we know that you need to attach the virus, but is that enough? You need to also support the virus to replicate inside the host cell, to produce more progeny virus, produce more babies, produce billions. So a single virus can go and produce millions virus in a single cell. Okay. Example is rabies virus. I'm giving a sideline view in this context. Rabies virus can produce 10 billion particles in a single cell if it is supported. Okay. So we call it, one is permissible, and it is that means it is going inside and replicating. To SARS-CoV-2 to replicate, it has also other drama to play around it. Because that it's not an easy thing for any virus to go. We have a dedicated immune system, well conserved and developed, and we have seen a lot of viruses, pathogens across our lifetime. And I don't want to go you in a detail. This is one of my published paper. What we are trying to tell you is that the AC2 receptor is engaged, and beyond that, there are like partner recognition molecules, toll-like receptors, then JAK-STAT pathway. So they all, work around to defend, not to allow the virus to grow. Then how can this virus grow? Well, the virus can easily grow because it brings with certain elements who can, counteract it. So if you can see, most of you who are undergraduate students, if you can see this is a sign, that means it inhibits or it stops. That means SARS-CoV-2 blocks interferon regulatory factor 3. Okay, so this is the essential factor which is going towards inducing interferon stimulated gene response. That means if the interferon is the innate response, immediate response. If you give a virus, there will be is there any questions at this moment, or shall I continue?
Sir, no problem, sir. Continue, sir. It's fine.
Okay. Yes. Right.
Yeah.
So this virus has to navigate through all these available molecules inside the host, block it, bypass it. It's like maze. Okay. So the COVID-2 has certain proteins. So spike protein, nucleic acid protein, matrix protein, polymerase protein. So these individual elements will play around it and go and stop this system to not work. Okay. And to be able to do that, and we know that the proteins of a mice cell will be different that from a bat cell or a monkey cell or a cat cell or a koala cell. So because the conservancy, as I showed you in case of AC2, similar pattern also is available here. So the virus has to go through this protein which can be blocked. Okay, that means a binding will stop it. If it doesn't bind, then it can't be stopped. So the host cell also provides the appropriate inner environment to grow the virus. So that is also essential to make a model system to exist. Okay. So here I'm just giving you that in addition to the model, virus now attached grow. What next? Next is that does it do the disease the same way what is being shown in human being? Okay. And for example, in case of SARS coronaviruses, we know that fever is a manifestation which is highly cytokine storm, we call it. Okay. Then beyond fever, we have disease worsens. Then we have this multi-organ problems, lungs, pneumonia, coagulation is one such example. Thrombotic events are happening where blood clotting is happening. So these are the associated conditions. How does the virus does that? Whether it is same in human being versus in the monkey. Okay, so we looked at the factors, and we could postulate long before last year that the major cause of the death would be associated with thrombotic events. And here we are showing that how the SARS virus is triggering or platelet factors such as complement 3 and C3A, B. I think these are the basic things which you have already studied in pharmacology and physiology courses. So this event should happen as exactly it should happen in the host. And for this, we cannot replicate this model inside the cell. It has to be in a system, in an organ system. The blood is not a cell alone. Liver is not a cell alone, or your lungs is not a cell alone. So that if somebody said, okay, we attach the virus, we grow the virus cell, then why you need animal? We can do in the cell culture. No, we cannot do it because we need to produce the disease in a model which will also show this kind of events. So that's the reason why we need a very, very typical system. So if somebody has to do those, work on the SARS, if somebody wants to understand the pathogenesis, whether they have to do the in vitro experiment or in vivo experiment. Suppose you want to do the in vivo experiment in the animals, then you need a large population, then you have to go for small. If you are going for a small population, you have to go for a monkey. Suppose you want to study something in transgenic mice, and you can use thousands or hundreds to your research, but you cannot do same thing in monkey, or you cannot do the same thing in ferrets or cats. These are regulated animals that we cannot use in thousands quantity. Okay, so you have to choose accordingly which model you have to choose. But if your job is not required animals, you can easily shift to the conditions very similar in the cell culture, which will allow the virus to enter and grow inside, do some effect, cytopath effect, and antibody testing. Previously speaker have told that the model, lab chip, this kind of information also easily you can extract without going to the animals. The choice is that what aspects of the viral pathogenesis you are looking for. Should you choose animal? Which animal? And if you choose in vitro, what kind of cells, what time, which whether we are 96 well, 380 well, or a chip or microchip, whatever the design of your study, plenty of choices are currently available. So choose accordingly what you need. So one important thing in the SARS coronavirus is it causes immunopathology. That means something is related to immune system is being manifested and it is causing the pathogenesis of disease. So the virus itself is doing something. In addition, our secretory molecules, MAP kinase, cytokines, CCL5, CLCXC, all variety of interferon, beta, alpha, all 16, they are also playing the role. Okay, so you have to, look at this population such as CD4 cell, B cell, T cell. All these things has to be in the place when you are looking for the immunopathology. So becoming very complex if you want to look at the antibody response, you want to look at the why certain vaccines works, sometimes vaccine does not work, why a patient is getting recurrent infection. Okay, so some of you have asked the question very recently that why the SARS, the detection is being not accurate. All these things are being hidden in this kind of study about the immunopathology. The rapid antigen testing, a plasma donor selection, all these aspects related to immunogenesis, and you have to be very careful that your immunopathological investigation should reflect what is being done in case of human being. So these are the background information where it gives you an idea that what to choose, when to choose, how to choose. Okay. So here, as I told you that the immunopathology is very strong. It causes cytokine storm. It induces coagulation. Okay, it does the granulation, degranulation of neutrophils, and activation of complement pathway, subsequently leading to the coagulation of blood. As the blood gets coagulated thicker, it cannot go to the brain, which are fenestrated with the blood vessels. As you know that the blood vessels in the brain is fenestrated. There is no lymphatic system in the brain compared to the lymph node as well as lungs and liver. So when the blood becomes thicker, it doesn't carry this necessary nutrients, oxygen, and organs die and organ failure happens. That is one of the major cause of the COVID after recovery, because the cascade starts even though patient is cured of virus. As such, there is no viruses or very little viruses. Because of this cascade of this cytokines and coagulation, people recovering from the virus coming to the home after hospital still and show this symptom. In addition to, I think the latest data is showing that somewhere between USA, the data I was showing in 15 to 30% of the people show the post-recovery microclots in their body. So that means this pathway is so accurately manifested that 1 in 3 person, probably, or 1 in 5 person, show some kind of microclotting blockage, the swelling in the leg or some kind of pain. So this is also a causative thing. So your animal model system also should reflect this amount of damage or this amount of activity which is going on the patients. Okay. So how to choose the animal model? Okay. What kind of animal model exists? For many virological research, we always assume, and not assume, we agree that all models are approximations. There is no exact thing. Okay. Nobody will mimic the human condition, and you cannot do the research in human in testing. Okay. So we have to choose the suitable model as per your need. What are the models available? Flies are available, eggs, chicken embryonic egg, or chicken chorioallantoic membrane, fish, mice, rats, dog, ferret, birds, non-human primates, research, macaque, all things are available. Some are vertebrates, some are not vertebrates. The lower the strata, it is easier to get a permission from your Institute Animal Ethics Committee. For example, if you want to work on fish or fly, it is less stringent. If you want to work mice or rats, a little bit more. If you want to work on dog, your Institute Animal Ethics Committee cannot take the decision. It has to go to the CPCC in Government of India, Ministry of Forest. Then they will give you permission. If you want to work on primates, non-human primates, such as research or langur or chimpanzee, so then you need another set of permissions, all these things. So depending on the stringency, the degree of the pathogenesis varies, the animal models also varies. So how to choose it? Okay. So the decision to choose the model based on a underlying principle that how does the host respond to the virus? Each host will respond differently. Suppose you will inject a SARS coronavirus to a chicken. What will happen? Okay. It will happen that chicken will take the virus. Very unlikely it will die. And while doing that, it will clear the virus and it will produce a beautiful antibody in that host. And that antibody will be neutralized. So if your aim is to look at the neutralizing antibody, where the antibody is binding to the SARS spike protein or somewhere else, why don't you use the chicken model? Why you are going for monkey? So that means that how does the host respond to the virus is the basis of things. Okay. Robustness. That means it should reproduce the again and again the same condition. Okay. Protective. That means you can contain that experimental conditions in an environment where it will not pass out to the other animals or outside the lab. It should not infect others. So this is how the animal endothelial cells, T cells, B cells, there should be plenty. There should be immune response. In addition to, you should also able to measure the outcome. For example, suppose you want to study the chicken cytokine response versus monkey cytokine response versus mouse. So we have ELISA plates available to know the cytokines, how much the quantity, quality-wise. But unfortunately, not all species cytokines can be measured in particular ELISA, because each cytokine, for example, let's pick interferon alpha. Interferon alpha is produced in human, in cattle, in bovine, in caprine, everywhere, any living organ vertebrates. But there are some degree of differences. So the reagents which are available in the market to study will not be suffice to cover all the animals' interferon. So you have to choose, and we know plenty that rat, mouse, cat, dog, bovine to a certain extent, they all are being available, but not all species are available. So you have to choose that measurable way, robustness, all these things you have to put into the table before deciding on that. Plus, are the animals are showing acute respiratory disease syndrome, which is the essence of the pathogenesis, systemic complication, multi-organ failure, death is happening. All these things you should look at it. In addition to, we also reflect the kinetics of the disease in the animals. For example, when we know that we are infected with corona, we are going for the PCR positive test, real-time PCR positive test. Some of you asked the question that can be real-time PCR give a false positive test? Of course, false positive test will be there. Real-time PCR is an approximation of light equivalent of a viral genome. What means that the machine looks at the signals in terms of fluorescence light. Okay, and that is the indicator of a presence of a RNA. So if there is a mismatch between these two, if the RNA cannot be reflected in terms of light, which is the fluorescence, whether you look at TACMAN versus cyber-green method, so which is another essential part I'm not going to talk. So there could be a false positive, false negative accordingly. Okay, so that's why we have control, internal control, which is the human genome, some housekeeping gene, external control, which is addition of certain RNA to that RT-PCR. What you add, you can see. Third is the virus RNA, which is the spike protein, nucleic acid protein, matrix protein, blah, 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 all these things. So unless these conditions are satisfied, a test is not test. We discard that test. We do not record the outcome of the test. So that's why there are stringency and all these things. I'm not going to talk that. But what I'm saying is that once we know that the RT-PCR result is positive, presence of RNA, then the symptom starts like fever, cough, dyspnea, respiratory disease, organ failures, occasionally death in human and all these things. So this is a kinetics of the disease, and prognosis is, as you know, that you can predict depending on the condition of the how the lungs is behaving, what is the multiple foci, or people do the C-reactive protein, and people look at the broad profile, people look at the presence quantity of viruses by real-time PCR. So all this predictability give you an assessment that what is happening in the host. So this exact same pattern should be reflected in the animal host. Okay. So what are the animal host? Okay, well, we have plenty of work ongoing. This is one of the biggest challenge in the modern history, modern virology beyond the HIV. HIV is the single most virus which has been studied since last 3 decades. Now the focus of the entire world is on corona. I need to tell you, but here we have to look at the animal models, looking at where you have to choose a large or small model, what kind of method you are introducing virus. So as we know that in case of corona, 99% cases the virus is contaminated or it transmitted through an aerosol route or through nose, not by eating something, okay, not by injection of something. In HIV, we know that sexual transmission, blood, and drug use is one such way, but not in case of corona. So you have to look at what is the route of infection and whether that can be replicated over and over and over. Okay, so whether you have to take a young population, you have to take a aged population. Okay. Example, some of you are from Nagpur and Madhya Pradesh, so Maharashtra. So you must have known about Chandipura viruses. Okay, one of the very fatal pathogen of human being, highly deadlier than Japanese encephalitis. Death rate is beyond 50%, but it causes death only in children. Adults are not susceptible. So that means you have to choose the model what disease is reflected. So earlier it was thought that SARS is very likely to do the condition in bad condition in the people with a higher age group or comorbid cases. Now that notion is going down. So SARS can infect even newborn babies unit. So as the progress of science information comes, your understanding on the virus is also changing. Accordingly, you have to choose what. Okay. You have to also reflect the same condition, like not necessarily always animals are diabetic, obese, okay, cardiovascular disease, infected with tuberculosis. All variety of things happen in the human society. Okay, so this same thing can be replicated in animals. That's the bigger question. Okay, so because unless we represent the not only host, we represent the spectrum of host, variety of pregnant women, diseased person, diabetic, obese, smokers, non-smokers, cardiovascular problems, having comorbidity with influenza or something. All variety of things, whatever is available in the human society, cancer patients should be also being reflected in animal model. So that is a bigger challenge. Okay? That's why we don't say that, when we do the vaccine trial, we choose the people for the phase 1, phase 2 based on the history. And for pharmacological people, and you know better than me, that when you do the enrollment of the subjects for a cohort, you have to carefully choose their condition, all these things. And we see a very efficacy 80%, 90%. But in real population, that efficacy changes because the vaccine trial group, phase 1, phase 2, phase 3, do not reflect the societal condition of us. Because we have to choose a population where in society we don't have a choice. Whatever available, we have to choose. That's why whenever there is a vaccine emergency authorization of vaccine happens, the guidelines come from the agencies such as CDC or Government of India. Okay, you avoid this, you don't do that, because the data is not there. Simply, whether it do good, bad, or good, that we don't know. But this particular vaccine is not tried in a pregnant animal, pregnant person. So let us think carefully, or in a comorbid person with tuberculosis. So these are the problems, when you look at the animal model, which will not reflect everything what we are looking for. Well, so as I told you in the beginning, that the ACE2 receptor in the rhesus macaque is the exact match what in human, followed by ferret, followed by all these things. So are they being used? Yes, people are using these models. Typically, for smaller works like antigenic response, immunogenicity response, we are using the mouse model or ferret model. For higher challenge studies, for vaccine study, we are going for the monkey model, which is rhesus macaque. In case of rhesus macaque, the histopathology of the organs very similar, and there is loss of appetite, complexity, dehydration, weight loss, symptoms in respiratory pain. All these things are being preserved in the human, what is being seen in nothing. Similarly, we have the ferret model. I'm not going to in detail, but showing you that how deliberately and diligently the community of science across the world are working and bringing to this information that what is what. Okay, so here I'm just showing you a monkey, ferret, human receptor expressing transgenic mice, and golden Syrian hamster. So these are some of the examples. This is not just 4. There are many such models. So when you are trying to do this animal modeling, you have to make a comparison. Okay, so comparison between the human versus that animal. So you have to look at the virus in the lung, particularly SARS. You have to look at the cytokines. You have to look at the damage. You have to also look at the infectivity in terms of clinical symptoms. All 4 columns should match. Then only we do. Okay. So here, can you do the same thing in mouse? So earlier mouse was not a subject of interest for corona, but there is something happened in 1983. We now make it 2 versions of mouse. One is mouse which are expressing human receptor of ACE2. So people can make nowadays transgenic mouse. They can copy the exact receptor of ACE2 which is present in human and make a transgenic mouse. That's why I call HAC2 mouse, which is Jackson Lab, got millions of dollars, and others. Charles River also, they got it a lot of money. Some of my colleagues in THSTI as well as in Institute of Life Science, NIV, they also borrowed and purchased those mice strains where they can do this modeling studies. So this is one such thing that mice are now available to do that. Beyond this, transgenic mice, we have a 3-decade history of humanized mouse model. So humanization in the sense here is that how much the organs and the physiological environment you can recreate inside the animal host and try to mimic that condition. So here, what people usually do that they take the animals, mice are the rats and the 2 models, and I'm talking about mice model. So they put certain doses of the UV light to deplete the bone marrow cells. So irradiation is happening. So that means your growing cells will die in response to the eradication. And once the growing cells die, your bone marrow is not having any good quality hematopoietic cells and stem cells. That time you are immunologically weak, and the animal is now cannot react, and it has a immunosuppressive environment. And that time you transplant fetal liver, human fetal liver, So patients are the babies, those are dying or aborted fetus. So there is a specific, ethical way we collect it and you transplant it, and then we follow that animals, and you look at 16 to 18 weeks, so that the human condition of blood, lymphoids, T cells, B cell, monocytes, everything is now populated inside the mouse, and that mouse is now called humanized mouse. Okay, so there are various approaches of humanized mouse. There are 3 major approaches. I'm not going to talk in detail, just going through that. And this particular system has revolutionized the HIV research. Okay. So looking at that, using this system, we used the antiretroviral therapy a lot. And nowadays, if you know that the HIV is less dangerous than diabetes. That means if somebody has HIV, we have so high active antiretroviral therapy, or we have this combination of antiretroviral therapy, that a person is losing only 5 years, okay, from its life. So that means if somebody has a virus, they can take the drugs or medicine. Only, if somebody is supposed to die at 85, will die at 80. But if the person is diabetic, he lose additional 7 or 8 years. So if you're in Delhi, your air pollution will take out 10 years of life. So to say that HIV is less dangerous than diabetes than air pollution in Delhi. So not to quote me in a particular manner, but this is the truth. Okay. How it is impossible? Because we made a situation where the humanized mice is available, because HIV was a very difficult virus to grow, and this humanized mice revolutionized the drug discovery, all these things. Nowadays, nobody is worried about HIV, although HIV is prevalent very heavily, pandemic. But are you worried? Even people don't think HIV every day now also, because so good quality antiviral molecules are available. So this is the beauty of this humanized animal model. So here, the first humanized animal model was like human peripheral blood lymphocytes. The liquid portion of the human blood was presented in the animals, and that was the exactly study purpose for the HIV, because we know that HIV has the T cell CD4 receptor and CCR5 receptor to entry. So this particular model really helped to look at it. And beyond that, this humanized model are also being used for innate immune response, B cell response, lymph nodes, other research, human immune system research, so hematopoietic stem cell research. So many ways these humanized mice are being used. Okay. And coming back to our context, can the humanized mice be used in case of HIV? What we saw, can it use in SARS? Of course, we can use in SARS. Okay. So we have this models are existing. Couple of companies, particularly Jackson and Charles Ribbels, are selling it high-priced this mice. Okay. In the past, we have shown HIV, Casimir forest disease virus, cytomegalovirus, Kaposi sarcoma virus, then Epstein-Barr virus. All these viruses have been used in this animal model, and this is a very beautiful model where you can look at skin, lungs, liver, all these things. Okay. Okay. So this models which is currently available is one step notch ahead, because this model in humanized model is already having a humanized receptor, which is SAC2, which is human SAC2 receptor in the transgenic animal. And top of that, there is the humanization. So this is a much better model than what was available to the HIV research. Okay. So we can do the challenge study, we can record the events, and subsequently we can look at it. So here I'm just giving you a broad view. I'm not going to read very texty and a lot of information here, but to show you that how this humanized mouse is producing the same amount of viruses what being done in other models, and the pathogenicity is replicated, and it's a beautiful model all across the globe to study this disease. Okay, so when you look at the SARS coronaviruses, the models are available: macaque, cat, ferret, hamster, mouse. Which one is good for you? What is bad? So that's certain advantages, disadvantages as usual in any system. Okay, so somewhere the virus will grow better, somewhere it will not. Somewhere the animal will kick out the virus very quickly in the innate response before the adaptive response comes. In some cases, the virus will stay longer, which is a kind of chronic infection. Some cases, the animal will lose more body weight compared to others. Okay, so like that, there are pros and cons in each model. I'm not going to discuss about it, but if you are looking forward, plenty of information and available in this domain. So now we talk about this, pathogenesis, that how the animals being used to study virus. A little bar review about vaccine development, and for a audience like you, and based on the previous speaker, we know that animals are essential for the vaccine research study. And not to emphasize further, and this period of animal models in preclinical studies unavoidable. That means we got to go to the animals before we got even start the phase 1 trial. So we have to use the basic immunogenicity study, we have to do the risk assessment, and we have to do the challenge study. So that means in animal model, you have to focus on immunogenicity. That means how much the spike protein, how much the virus is required, how long, what is the dose interval. All this basic information comes from this preclinical studies. Similarly, whether this particular vaccine is safe or not, that is also assessed in the animals. And challenge study. Suppose you give the vaccines and you challenge with deadly virus of corona, will the animal is protected or not? In simple terms, we have to satisfy these things, then only it is go to the phase 1 trial. The two topmost models which is being currently studied for this kind of work in vaccines particularly are the mice and monkeys, and we know that about these things. I'm not going to talk detail. Okay, so there are vaccines which are being DNA vaccines which Intas is handling and others companies are handling. They are using research macaque model. The Chadox vaccine, which is the name is Covishield, chimpanzee adenovirus Oxford 1, we call Chadox vaccine, okay, which is the Serum Institute of India in Pune is making it. So that is also tested in macaque and is mice. Inactivated vaccine, which Covaxin is making in Bharat Biotech. They are also tested in Wisconsin Madison in macaque model. And RNA vaccine, DNA vaccine, all variety of vaccines are being tested. And preclinical phase 1 is going on. In many cases, things are being already in the market and it is introduced. So you know that how essential the model is. Similarly, the drug discovery is also has a major role in looking at the animal models, particularly SARS. It takes years of research, decades of research to find a molecule, and I don't want to go in detail. But nowadays we have this computational biological approach. Schrödinger, for example, one software we can look at the synthesis of molecule. We can look at the cytokine response in the chips, which lab chips, which ma'am has told you in the beginning. So all these things can complement the animal work, not replace it. Okay, remember it. Okay, so and current we can do the in silico studies. We can make a molecule suitably stop the spike protein. We can look at the efficiency in the animal models. We can go to the different pathways and inject it, look around it. Okay. Beyond that, we can see that the remdesivir, flavipipir, hydrochloroquine, all these molecules which are being currently tried. Ivermectin is for a good example. So from my veterinary career, I was using ivermectin since last 30 years, and now it started being impacted to the human being. Ivermectin is a broad spectrum endo and ectoparasitis drug, and nobody knows how exactly it does the work for the SARS-CoV-2, but it appears that it has also mild regulatory role, and it augments the immune system. As a result, whatever the ways they proposed for the hydrochloroquine or CQ is a similar way it is playing some role in case of coronaviruses, and that was tested in a lot of animals. So flavipipir, is once a remdesivir, all these things tested in animal models, because they are rediscovered drugs. They are already approved by certain regulatory agencies such as FDA. Safety, efficacy is tested, safety is tested, redesigned, rediscovered, repurposed, now being used in the market or for the clinical use based on the animal data. Then I'll conclude my talk with the last part, which is the animals models for the studying genotypes. Okay. So nowadays there is a lot of political debate. Where did the COVID come from? Where was it? Okay. Is it the China made it in the one laboratory, or somebody introduced as a bioweapon? All this variety of things are going on. Okay. But at least we know that in 2003 there was the SARS-1, which is SARS coronavirus, was being identified, and in ferret model, and in 2013 the MERS, Middle Eastern Respiratory Syndrome virus, was identified. It took 10 years to know the origin of the virus in case of SARS, but 10 months in case of MERS, because there was a smoking gun. That means people could recover the, those who are infected with MERS, they could correlate easily to the camel, which are restricted in Saudi Arabia because population is low. So they could identify easily that MERS is originated from camel. But 10 years took for the SARS-1, and we don't know what is the fate of this coronavirus 2 or COVID-19 virus or SARS-CoV-2. Okay. So a debate is going on in a particular context. We always heard this word called gain of function study. Okay. So this is an ethical question that why we do this gain of function study in animals. Okay. What it achieves, what questions it makes, what kind of security it poses to the audiences. And I'll give you an example of that in next slide. But gain of function is basically that, we want to suppose you have a bike which is running 70 liters per kilo, 70 kilometer per liter of petrol. Can it increase to 100 kilometer per liter? That is called gain in function. Or you can lift 100 kilo in your two hands. Can you lift 200 kilo? Can you increase your capacity? So, which sounds good, but can the virus do the same thing? Can a virus kills only 2% and it will gain the function, it will kill 20% of the population? Each virus, modality, morbidity and mortality are different. Only one virus kills 100% is the rabies. Okay. Not a single virus is close enough to rabies. And we all know that rabies is the only virus which can be prevented 100%. That means if somebody also bitten by dog, if vaccines are given, even after post-bite, the animals can be, or the human being can be protected. So remember, the contrast nature. Okay. This is the only vaccine available after the virus goes to the body. And it is the only virus which causes the death 100% if vaccine is not given. So this kind of information we gathered from the animal experiment by gain of function. But unfortunately, this has been, being done in a condition that creates a lot of anxiety in the society, policymakers, countries, suspicion, bioterrorism, all this are coming. Okay. So what the gain of function people do that? So people do a serial passage of a particular virus. Suppose an animal died of a virus, rabies, for example. You pick the animals which is dead, you pass to another animal, that will dead, that will dead, that will dead. Slowly, you will choose a population of virus which is bigger and healthier, stronger in killing the animals. So these are the problems with the thing. And here I'm giving you an example where I was working, and my colleague Subhash Das, which is the fourth author in this paper, they are doing some kind of animal gain of function studies for the influenza deadly virus. Okay, so what they did that. So this was happening in Wisconsin Madison in Kawauka lab, very famous lab. Okay, and Subhash is a veterinarian. So they are testing the ferret model. Ferret is a very similar rodent compared to animals, but it is a better model because it provides the respiratory pneumonic condition like influenza in these things. So what Subhash did that. So he simply took some viruses, injected to a group of animals. 10 animals are there, sprayed it like aerosol, just spray. And he took look at the animals who died fast, he picked that animal. He relieved the other animal who died fast. That means there is some virus which is very dangerous in that animal. Then he collected the virus from that animal and sprayed to another group of 10 animals. And then who dies first, then he picked. So like that, he did 10 rounds of re-enucleation, reintroduction of the viruses. And at the end, he got one kind of viruses which is very strong and kills 100%. That is the way people do the gain of function for pathogenesis. But in other way, you looked at, you choose the animal which is not dying. Then you collect the virus, give to another animal which is not dying. So in that way also, you select a virus which is attenuated, can be a vaccine. So both ways you can look at it. Okay, so it's still a double-edged sword. When Subhash, try to publish the work, they send it to, Science magazine because that Science and Nature are the two competing group, very famous group for journal publication. Science is being governed by the University of California and group. They immediately flagged to the FBI in USA, and FBI closed the lab. Not only that, there are a lot of discussion in CNN, debate on listing that this, the money is being spent by taxpayers in government of USA. People are misusing, making very deadly viruses, bad for the society. The situation becomes so worse that all the researchers in the country in USA gave up doing research in influenza for 3 years, called moratorium of research. Okay, so you can understand the gravity of the situation. Science never allowed to publish the articles, but as a result, there is a competition going on. Nature took it, and this article was published in Nature. And this is the story I can say you that how these things, animal models can do these things. So this is last first one slide. And in a simple slide, I told you that starting from wildlife, pathogenesis, emerging diseases, domestic animals, viruses are everywhere. Whether it's Ebola, SARS, COVID-19, rabies, they are circulating, recirculating. We are the only species we are being cut off by this nature. It is there. It is there in nature. Okay, and evolution is making such a way that the fittest is again surviving, and viruses are there, animals are there. It is existing, coexisting in the nature. Only thing that we are not fit enough to handle the virus, and that kind of aspect we need to know more by looking at animals and nature. Okay, to conclude that animals are indispensable for any modern virology research, and we should focus on minimizing research of the animals. And all models are approximation. Not a single model is exact. And I just quote another famous virologist, Vincent Racagnola, that whenever you do research, the mice lie, monkey exaggerate, and ferrets are not human. So choice is yours now. Okay. With that, I invite your questions and let us discuss.
So thank you very much for your nice presentations today, sir, because that is a very important thing for everyone who are the research scholars or who are the upcoming scientists. And it is very essential to know the pathogenesis as well as the transmission of the disease. Once we will get the idea about the pathogenesis or the mechanism of the disease, then we will going to target for their antiviral effect or target for the antiviral treatment, sir. Thank you very much for your nice presentation. Then shall we, sir, proceed for the questioning answer center?
Yes, please.
Okay, sir. Thank you. So now the session is open for the question answer. Yes, if any participants have the questions, please write in chat box. Okay, we will forward this question toward the sir. Okay. So, sir, the one question is there, sir. What are the difficulties for the development of the antiviral drug for the treatment of the corona?
Okay, so corona has surfaced since 1 year. Typically, the antiviral research takes a longer time because we have to hit and try method because it's not a straightforward method. Okay, so problem that there are two components of the virology. One is prevention, one is cure. Okay, what happens in the long run? Prevention is cheaper. Okay, in the sense that if you buy a vaccine for rabies, it costs only 100 rupees. But if you buy remdesivir, it costs 10,000 rupees. So it was not incentivized for a broader scale for the private companies and government to invest on this particular domain that produce more antiviral molecules than vaccines. Vaccines was easy. Okay, so otherwise the field was not progressed in one manner. Second is that because of the new viruses and because of the corona was wiped out for long run, people did not invest in learning that. Rather, they invested in Ebola, Zika virus, or something which was economically or commercially exciting. So that's why no antiviral is available at this moment. Only you are using Ebola or influenza kind of antivirals to repurpose in case of corona. So this is why the delay is happening. But in future it will come.
Okay, sir. Thank you. The second more question is there, sir. Is there the pathogenesis and the transmission is similar for animals as well as the humans? Is similar, or is there any difference?
No, not
pathogenesis. Pathogenesis
yes.
of this COVID-19.
So when you look at pathogenesis, there are two angles. One is a cellular response, other is a host response. Okay. So cellular response is very similar. That means how the virus goes, how it go to the nucleus, cytoplasma, made the protein get translated, they are glycosylated. They are 99% similar. But the problem happens with the host response, like beyond the cell, the cytokines. Okay, the damage to the lungs. Okay, example, a simple example is your malaria. Okay, malaria will kill you, but it never kills the so-called mosquito. Okay, chikungunya virus kills the human being. So degree of pathogens depends on the kind of the conservancy of our certain elements or genes between that particular animal versus human being. Okay, monkey versus human being or mice versus human being. To more extent, we are trying to make as much as possible to the human oriented, but it is not the same. There is a gap.
Okay, thank you. Sir, if the animal model is not exact how the pathogen is, then how we will going to correlate that models with the human model? So
that's what I mean, that is the approximation. So we live in the world of probabilities. Okay, so number of probabilities. Okay, for example, if you get vaccinated, what is the probability you are being hospitalized versus you are not having vaccine, and what is the chance? Here is that if you don't get vaccine, chance is that you'll get 1 in 200, but if you get vaccinated, you'll be 1 in 5,000. Okay, so this kind of modeling, that's what I'm trying to tell you. This kind of information is extracted based on the very many parameters. For example, a cytokine is produced, antibody is produced, the neutralizing antibody is produced, the period of the disease staying in the animal, the recovery rate, the death rate, the loss of body weight. All these parameters will provide you information, the probability of a human thing. So based on that, whether we take the call or not. Okay, so based on the relationship.
Okay. Thank you. Sir, so this question is other than the COVID, sir, which is the ideal model for the toxicity study of the various drugs?
Sorry, which is the ideal model for
toxicity study of drugs?
progressivity study.
Toxicity.
Oh, toxicity, sorry. Yes.
Yeah.
So usually toxicity studies are done in only rabbits and mice. Okay, because we look at the liver, lymph node, kidney, lungs as the indicator of the toxicity, accumulation of the cells there, and pathophysiology. So for toxicity aspects, we do that study. And before that, nowadays people are looking at the cytokine response and all variety of things in animals. So we don't go for the human for toxicity. Rather, we restrict in rabbit and mice alone, not in monkey also.
Okay. So rabbit is ideal for the toxicity.
Yeah, that is.
Okay. Thank you, sir. Thank you very much, sir. Sir, another one question is there, sir. So what are the reasons or what are the factors for the development of the new strains of the SARS-CoV-2?
I'll give you an example.
Any reason? Is there any man-made reasons, or it may be the natural thing?
So I think I showed you the example of Subhas Bal, that he was simply taking virus, passing through a set of animals. Yes. Animals were dying first. Don't you think that this is not magic? We can also do the same thing if you are allowed to do that. And there are some countries, some laboratories are also notorious. They're doing it, not now. The existence of this, so-called bioweapon, which is 50 years of concept, is going on. Okay, we don't know. We are just knowing. That is one aspect, which is the artificial way of manipulating. The other is that the antibody escape or the immune escape. So what is happening that there is an interplay between me and my virus. Okay, I try to fight with it. Okay, some point I will mount the pressure such a way that the virus will go down. Okay, but the nature has made the virus such a way that it will create a quasi-species. It will make a mutation of itself. Okay, it changes so rapidly in a matter of 30 minutes. If one escape out of million, that will become a new strain. Okay, so that is the where the problem lies. That the constant change in the virus genome, because of the problem in the virus polymerase or the replication system, it is not a foolproof. It will not replicate exactly as the copy. We human beings can replicate very easily, but the virus will do purposely mutation, and it is good. 99% of the cases, the viruses die. 99% of the quasi-species are not even infective, but we don't need 99. We need only one to come out and do the nonsense. So that is the whole problem with most of the RNA viruses, including corona and HIV.
Okay, sir. Thank you, sir. One last question, sir. Sir, how many days the protection from the corona vaccine after the booster dose?
Just now we did a research study with Armed Force Medical College in Pune. I think all of you know it. So they gave us 400 samples from the cohort. Okay, so we know the cohort means. So one set of sample is that people, those who never got infection, vaccination 1, vaccination 2. People got vaccine, but infection, and people got infection, no vaccine. Okay, 3 types of situation. What we noticed now, this is very relevant data, and we are going to publish it, and probably it will be the stepstone towards the guideline. That 3 to 4 months is the latest period. We received the serum up to 128 days, because they got the vaccine in January and the serum came in May. It is giving a very high neutralizing antibody till 4 months, and we don't know the data after 4 months. Those who have been vaccinated once, okay, and we don't see the significant change in the quality if you get the booster. So when you say the quality, means that the variety of strains are available. The Delta strain, B11 strain. So if you are immune to all these strains, that is called quality. Okay, so whether you get 1 vaccine or 2 vaccines, your antibody is being same for the both cases, quality-wise. But quantity-wise, there is some changes. And that changes mean that if you get a booster dose, you will have more antibody in the body, but not necessarily that antibody will be neutralizing all viruses. Whatever you do in the first booster, same thing will do in a rapid manner. And in North Cell, the data is very encouraging to tell you that it is, in one shot is good enough for at least 5 to 6 months in my view. 2 shots should be good enough for a year. That's the current understanding. But we don't have a data, we don't have a samples yet because the study is ongoing. And only in January, we only got the vaccination. So my view is more optimistic than what is being told to the media and all. I see that very high quality antibody, up to 2000 dilution, which is very strong, okay, is coming up. These are real patients.
Okay, sir, thank you. Due to the running of time, it is not possible to take all the questions. All the delegates, all the research faculty, or the research scholars and the participants are requested to write the question in chat box. We will going to communicate with the sir, and sir will mail all the answer for you. Yes, with a thank you very much, sir.
Thank you.
At the end,
thank you.
Thank you very much, sir, for your motivational and inspirational speech on the topic of animal models for SARS and COVID-2 pathogenesis in transmissions. So that will definitely helpful for the upcoming scientists or the upcoming research scholar for making the target for the development of the antiviral drugs, or it may be for the development of the various vaccines also. Yes, we are very much thankful, sir, for accepting our invitation within a very short span of time. Thank you very much, sir. On the behalf of management and the principal, I would like to just extend a warm gratitude towards you, sir. Thank you very much.
Thank you all. Thank you.
Thanks.
Bye-bye.
So all the delegates and the faculty members and the research scholars are hereby informed that within a few seconds, the next session will going to start. Stay tuned. Okay. Now the session is over to Kulkarni, sir. Kulkarni, sir.
Thank you, Vadukar, sir. After this brief session by Dr. Devashish Naik, I want to introduce you, and I feel this immense pleasure to introduce you our today's speaker of this session, Dr. Rahul Kodugu, on topic efficacy and safety of COVID-19 vaccine. He has completed his MBBS from Government Medical College and Hospital, Aurangabad. He has done his postgraduate diploma epidemiology from Public Health Foundation of India. And publication side, he has published numerous papers in journals of international review like Pharmacological Therapeutics, Current Opinion, Allergy, Clinical Immunology, JAPI, American Journal of Respiratory Critical Care Medicine, and Journal of Postgraduate Medicine, to name a few. He has received a total grant of US dollars $250,000 from National Institute of Health, NIH America, and Vodafone Americas. He has presented his research work in many conferences which are held at Barcelona, Amsterdam. He has received a gold sponsorship award from European Respiratory at ERS Congress, Amsterdam. Also, he received silver sponsorship travel award from European Respiratory Society. He was a young scientist awardee at Napan Bhubaneswar. As a clinical trial investigator, he has worked on 16 clinical trials in asthma and COPD. As an invited speaker, he has delivered talks at conferences and workshops held at MIT Boston, USA, Tanzania, International Conference on Pulmonary Disease, Intensive Care and TB, Tehran, Iran, and at Manipal, Akinova. On professional experience front, he has worked as a medical officer in Ahmednagar and as short service commissioner medical officer at Army Medical Corps, Indian Army. He has worked as a head academic research and director, Advanced Pulmonary Function Testing Program at Chest Research Foundation, Pune. And presently, he is working as a general manager, Global Clinical Development, Glenmark Pharmaceuticals Limited, Mumbai. With this brief introduction, I would like to hand over this session to Dr. Rahul Kodugu. Dr. Rahul Kodugu, you can proceed for the session.
Thank you very much. Can you hear me?
Yeah, we can hear you, sir.
Yeah, I am unable to share my screen.
Sir, you can go to share screen.
Yeah, but I think I have to be made co-host for that.
Sir, you are already co-host, sir. The permission is granted to you.
So here it says host disabled participant screen sharing. So I have joined from 2 accounts from where my video is
Acha, what is the name of your account, sir, then?
Rahul Kodgule laptop. So that account you have to make host.
Acha. Acha, Rahul Kodgule laptop. Okay. Please be online, sir. Now please try once.
Yes, I can share now.
Thank you.
Yes, thank you very much for that wonderful introduction and for inviting me for this presentation. So I thank the organizers for organizing this important scientific event where we get to discuss some of the aspects related to COVID-19, and it's helpful for everyone. So it's a learning for me as well as for everyone in in this meeting. So what I'm going to do in next 30-40 minutes is I'm going to speak about the efficacy and safety of COVID-19 vaccines in general. So I won't be specific about certain vaccines. I will take 1 or 2 examples where required, but my presentation is mainly focused on in general understanding of how vaccines are developed and how do we know whether they are efficacious and whether they are safe enough to be used in community. So recently we have every day there are headlines related to COVID-19, and a lot of them nowadays are related to vaccines, and we have these issues of Oxford vaccine sometimes being not accepted, sometimes being accepted. So there is confusion among population about whether the vaccines are really safe and whether they are efficacious. We have news about some vaccines claiming 90-95% efficacy, some vaccines claiming 60, 70, 80% efficacy. So are some vaccines better than other vaccines? So there are many such questions that we will try to address in this presentation. So these are the several efficacy rates that are there in public domain. So vaccines efficacy ranging from 66% to 95%, and this gives a picture that we think that some vaccines are better than other vaccines. And is that really true? Can we actually compare these efficacy rates between vaccines? And we'll try to understand that in this presentation. But overall, if we look at the efficacy rates, these are actually very encouraging. If we see the efficacy rates of vaccines before COVID-19 vaccines, like influenza vaccines and other vaccines, the efficacy rates were very low. And here we are getting very high efficacy rates. So why is that difference? And how today's vaccines are actually better, probably better than previous vaccines. So we'll see all that. So let's go briefly through the history of vaccine development. So in 1796, Edward Jenner developed the first vaccine, documented first vaccine for smallpox. And it's been more than 200 years since then that vaccine development has been going on. Initially, it was at a very slow pace. The next vaccine came in 1880. Then almost after 40 years after that, the next vaccine came. So the vaccine development in the initial period was very slow, but in last 30-40 years, many vaccines have come. So most of the vaccines that are available today have come in last, let's say, 50 years. And currently there are more than 100 vaccines, much more than 100 vaccines that are approved globally for various infections. What is important is all that development that happened over more than 200 years is now happening in just 2 years, year 2020 and 21. There are more than 100 vaccines that are currently in development only for one disease, that is COVID-19. And more than 50 vaccines are currently in clinical trials. So much research in vaccines has never happened. So it's unprecedented. And what is happening today, a lot of these things are new and happening for the first time in the world. And that's quite encouraging. So we have had some previous talk about the immune response. So I will just try to summarize how immune response takes place when there is a virus infection. So this is just an example of avian influenza infection, which is not very different. So it's like a viral infection only. So when there is a viral infection, the first line of defense is the innate immune response. So if it's a respiratory virus, so the respiratory epithelial cells and the local inflammatory cells will try to kill that virus or destroy that virus. And quite often than not, this is successful in day-to-day life. And we are able to get rid, there are always viruses in environment, and we are able to get rid of those viruses based on innate immune response. Sometimes the innate immune response fails, and that typically happens when the person is weak, the immunity is weak, or that virus is strong enough. Virus has a high infectivity, and virus also has a propensity to cause more disease. So in those situations, the local innate immune response may not be adequate, and then that leads to the acquired immune response, which is mainly in the form of T cells and B cells. And here we can see the majorly the B cell immune response that takes place, and also the T cell immune response, mainly in the form of cytotoxic T cells. So when virus infects these cells, they remain in the cell. So it's predominantly the cytotoxic response that is important to get rid of these viruses. But over a period of time, the T cells, there are also CD4 helper cells which create an immune response which helps the generation of more antibodies. So antibodies also play a very important role against in the immunity against the virus. Although so far it's believed that it's a cytotoxic response that is much more critical. Now, why this is important is because when we talk about vaccines, vaccines typically have been dependent more on the B cell or the antibody response to the virus, while the natural ability of our body is to respond mainly in the cytotoxic form. Although vaccination also leads to some cytotoxic response, but so far the evaluations and discussions have been more on antibody response, probably because it's much easier to study antibody response. And that leads to some difference in terms of natural immunity versus immunization. Also, there is one important difference that we need to understand is when there is a respiratory infection, it's a local infection. So in the beginning, so it starts from the nose and upper respiratory tract, goes to the lower respiratory tract. While the vaccines that we give are mostly systemic, so they are mainly intramuscular or IV vaccines. So the responses may be different because of that also. So there is some difference between a natural response and immunity generated by a vaccine, and we need to be aware of that. So let us look at the vaccine development process. So this is the typical process that has been followed over last 50 years, and here we can see that takes about more than 10 years, about 10 to 15 years to bring out a vaccine. But in this pandemic, we are bringing vaccine in less than 2 years. So how is that possible? Are we compromising somewhere? Are these vaccine shortcut vaccines which may have damage? So we'll try to address some of these questions. We'll just divide the time, what takes a lot of time. And this is the initial process that takes a lot of time. So discovery and target validation, preclinical stage, and manufacturing and development. And these things actually take a lot of time because some of the virus infections are not as common as COVID-19 is. So it's to isolate the virus, to study the virus, to study the antigens on the virus. It takes a lot of time. Because COVID-19 is a global pandemic, right from the beginning, right from the time this virus spread to all over the world, since that time, a lot of research has happened, and every day there are multiple publications coming out. So as a result, this process that takes 3 to 2 years time actually happened in a couple of months. awareness about the virus, a lot of information about the virus became available in just few months after the virus spread all over the world. And that helped shorten this first phase to a very short time. So once we know the structure of the virus, we know what proteins in the virus are important. We know that spike protein is the most important part in terms of leading to infection and also for vaccine development. So that knowledge came very quickly in this pandemic. And then going to the preclinical stage is also has been fast. And that's mainly because there have been a couple of viral infection in last 20 years. And the SARS-CoV coronavirus 1 infection, which happened recently, a couple of years back or a decade back, vaccines were being developed for those infections also. So whatever knowledge and formulation requirements were there, they were already, people were already available. The vaccine industry was already aware of how to formulate a vaccine. Even preclinical studies were done with the SARS coronavirus 1. So even that knowledge was available. So all that helped to fast track the process. So there was no compromise made anywhere, but this whole process, it was possible to do that within a span of few months. Then coming to the clinical phase, also there is clinical assay optimization, which took some time. It does take some time, but because of the previous knowledge, even that could be done very fast. And then the clinical phase, which takes about 2 to 10 years. And mainly the time taken is because these studies are typically done sequentially, phase 1 followed by phase 2 followed by phase 3. And there is always some gap between the two studies because of regulatory evaluation, data generation and all that. And biggest problem is the availability of or the incidence rate or the event rate of being positive, converting to say infection positive patient. So that happens very slow for a lot of diseases that are not so common. But because this COVID-19 is a pandemic, there are so many COVID positive cases coming up every day. We have lakhs of them coming in India every day. So with that number of cases, we don't have to wait for years to get the results of the study. In few months, we are able to get the number of cases that are required to demonstrate the efficacy of a vaccine. And that also helped a lot. And so here again, what I want to emphasize is, even in this part of the development, there was no compromise made with the quality of the data that was being generated and even the quantity of data. Because of this urgent situation, some of the studies happened in parallel instead of sequential. And so data requirements were made little less stringent. So, and without compromising with the efficacy and safety. So all this allowed to bring so many vaccines in within a span of 2 years. Even regulatory review, you can see here, takes about 1 to 2 years, and that was also done in a period of weeks. So all that thing helped to bring these vaccines very quickly to the market. So let's see a little bit about the SARS-CoV coronavirus 2 virus and or any coronavirus. What typically happens? Some of this has been discussed in the previous by the previous speakers. So important thing here is the spike protein. So here we can see this is a virus and this has a spike proteins. This spike protein is critical for the virus to make an entry into the cell, into a human cell. Without this protein, the virus cannot cause an infection. So this spike protein is very important. And there is something, this part of the protein that binds to the receptor called as a receptor binding domain. So within the spike protein, that's the most important part of this coronavirus. So if we can make that part ineffective, this whole process of infection can be avoided. And that has been the basis of developing vaccines for COVID-19. So all the vaccines that are being developed currently or that have already been developed, they are targeting this part of the coronavirus. And some vaccines are whole virion, some are targeting only the messenger RNA. But basically, all of them are effective against the most important part of the virus, and that's why uh, it's it's not expected that there will be a big difference between the efficacy and safety of these viruses. Of course, whole virion virus may have efficacy against various antigens because even if there is a mutation, so what is expected is because we are taking the whole virion, it may have some protection against other mutations also. But then it has to be balanced with safety also. There may be safety issues also with the whole virion. So, and these differences also are very minor. So, in nutshell, it does not appear that there will be any major difference between the different vaccines that are being developed. So these are the several strategies that are being used currently for the development of vaccines for COVID-19. So from messenger RNA to the inactivated or live attenuated virus. So all these strategies are being used, and these strategies are not new. They have been known, except for the mRNA. All the other vaccines have been tried and known to be successful, and mRNA has a very strong reason for being successful. So as we discussed the vaccine development pathway, the first things after the lab experiments comes is the animal study. And what typically happens here is we have identified this receptor binding domain as the most important antigen for developing vaccines. And it's a part of the spike protein. So different companies will have different ways of formulating this vaccine. So they will use different excipients. Alum precipitation is a common and popular method, but there can be different methods. And they end up developing a vaccine. And this vaccine is then given to, it's first tested in animals. So in animals, these vaccines are given and the animals are exposed to the virus, and the response is seen in terms of how many animals who are vaccinated get infected compared to how many of them who are not vaccinated get infected. And that gives the confidence of whether the vaccine that has been formulated actually works or not. So even before coming to humans, we know a lot about a vaccine's efficacy and safety. So different doses and different formulations usually are tested in these animals, and the best candidate is then taken forward for the human development. So even before coming to humans, there is a strong confidence about the efficacy and safety of a vaccine. So when it comes to human trials, what actually happens is there are three typical phases, as we know: phase 1, phase 2, and phase 3. And the phase 1, in about 100 people or in less than 100 people, these vaccines are tested mainly from the point of view of safety, because you have tested in animals. There may be some differences between animals and humans, which we don't know. And that's why it's important to establish the initial safety in humans, in healthy volunteers, where these vaccines are given, and mainly it's the safety that is evaluated. And this safety is not evaluated just in terms of giving a vaccine and seeing whether this patient has any adverse event or any symptoms or any problems. The evaluation is much more thorough. It involves complete evaluation of the organ systems by doing several detailed tests: detailed liver profile testing, renal profile testing, detailed ECG continuous monitoring of the heart activity, and so many other tests are done just to make sure that none of these parameters we get a signal about the safety of these vaccines. And it's only when in this phase 1 no significant safety signal is found, only then these vaccines go to phase 2 trial. In phase 2 trial, there may be several objectives. It's a little different than the drug development. Vaccine development is slightly different than that. But it is in this phase where we try to have some initial impression about the efficacy of the vaccine, mainly in terms of the seroconversion or antibody response. So if this vaccine actually works, then we do expect to see seroconversion or some antibody response after vaccination. And also it's also in a larger sample, we get some idea about the safety. And here so in phase 1, we typically take very healthy people, but in phase 2, we try to take more general population of different age groups, sometimes even older people. We try to balance male-female ratio, different demographics. We try to include as many people from lower socioeconomic strata, higher socioeconomic strata, to make this phase 2 little more reflective of what we are going to see in phase 3 or in general population. So this is a little robust evidence of safety as well as some evidence of efficacy. And from there, the vaccines go to the phase 3, where studies usually are supposed to be large, in a sample size of 30,000 or more subjects, who are of which half of them may get vaccinated and half not vaccinated, or little less not vaccinated, to see the actual efficacy of the vaccine. And efficacy of vaccine typically is defined as, after giving a vaccine over a period of time, how many patients uh, are protected from infection. So that means how many patients do not turn positive compared to patients or subjects who are not given vaccine, how many of them turn out to be positive. So based on that, efficacy is calculated for COVID-19. Globally, regulatory agencies and WHO have added some more measures as an option to demonstrate efficacy, in terms of whether the vaccine prevents a serious infection, whether the vaccine prevents hospitalization or ICU admission, or whether it prevents mortality. So all these are a part of evaluating efficacy of a vaccine. And obviously, safety is another very important aspect of vaccine trials. Uh, yeah. So this is in general how vaccines are evaluated. Uh, uh, in the phase 2, forgot to mention, in the phase 1 and phase 2 also, we get a fair idea about how the regimen of vaccine should be, whether a single dose is going to be enough or will require multiple doses, and at what should be the difference in duration between these two doses. So all the vaccines that are currently authorized, these are not, we cannot say that these are approved vaccines, because a lot of studies are ongoing and some more data will be required. But these have received emergency use authorization, which means that the data indicates that there is no common or there is no important safety finding from this study. Something that will not allow mass vaccination. So such safety findings haven't been found with any of the vaccines that are given emergency use authorization. And also there is enough data to show that these vaccines provide adequate efficacy. So the cutoff that WHO has for adequate efficacy is at least 50%. So at least 50% of the people should be protected from infection. Only then an emergency use authorization is usually given for COVID-19, or at least WHO would recommend. So now we will come to some of the data, some specific data about some of the vaccines. I don't claim that a vaccine is better than any other vaccine. So we will see why, but we'll see some data of what work has happened and how these studies have been conducted. So this is the phase 2 data of Covaxin that was published in Lancet Infectious Disease. It's an Indian vaccine, a whole virion vaccine. And this is the patient flow. If you look at, so 921 patients were eligible and 380 actually were randomized in the study. 190 received 3 microgram dose of vaccine, and 190 received 6 microgram dose of vaccine. It's a phase 2 study. What we are more interested in seeing here is the antibody response or seroconversion. So here, if we look at the population that was taken, so 90% subjects were between 18 to 55, about 4% more than 55 years age. We know that at older age groups, the antibody response may not be very effective. And maybe that's the reason why there are less people in this age group. But we'll have to see actually how antibody response is there in that age group also, because that's the age group more than 65 mainly, where the need of vaccine is the highest. And male, female, majorly the subjects were male, but there was a fair representation of females in this population. And these are the results. Here we can see that. So this is the time axis: 28 days, 42 days, 56 days, and this is the wild type neutralizing protein response. So here, basically, the antibody response we can see. So over a period of time, there was a good response in antibody with both the doses, with 3 microgram and 6 microgram doses. No major difference between the two doses. So this is 3 microgram, this is 6 microgram. And this is the seroconversion rate. So seroconversion in this study was defined as the antibody levels more than 4 times of the baseline. So large proportion of patients, so after the first dose, so at day 28, around 39-47% subjects, they had a seroconversion. But after the second dose, at day 42, day 56, at day 56, close to 100%, around 98% subjects had seroconversion. So this is a very clear evidence that this vaccine works. Now, this antibody conversion does not mean that that's the efficacy of the vaccine. Efficacy mainly is in terms of how many subjects can be prevented from having infection. So this does not truly reflect, antibody levels also do not truly reflect the severity of disease that a subject may have. So if someone has high antibody levels, does not mean that this person will have a mild infection or a mild disease if this subject gets infected. So it's not totally dependent on the antibody response, but it gives a very good, clear idea that large number of people will be protected against COVID-19 disease if they are vaccinated by this vaccine. So these are the actually same results in numbers. So if you look at the antibody response at day 56, it's 6 microgram dose about 97, with 3 microgram dose again around 97%. So very good antibody response has been shown with this vaccine, and similar data is there with a lot of other vaccines, although not all data is in public domain. So I used this just as an example, but nearly all vaccines have good efficacy. So this is the TH1 versus TH2 positive ratio. So we saw that cytotoxic T cells play a very important role in viral infections, and that reflects mainly the TH1. So here we can see there is some balance shifting towards TH1, so which is a good thing, which suggests that this vaccine will have good effect against preventing infection. And similar things are reflected here. So significant increase in interferon gamma and TNF alpha, suggesting a predominantly TH1 response. So this also is a good news for the vaccine. Now we'll see an example of the phase 3 study. So this is a Sputnik V vaccine. So I've just taken this as an example. I'm not suggesting whether this vaccine is better or worse than other vaccine, but this is just an example to how we should look at the vaccine data to ourselves understand which vaccine is better or not. So here we can see the population. So around 22,000 subjects were randomized. So roughly we saw that earlier that typically the studies will have sample size of 30,000 or more. But because this COVID infection is so predominant, even lesser sample sizes will give us almost the same information that larger sample sizes will give. So here these subjects were randomized and they were given 2 doses of the Sputnik V vaccine. So these are the baseline characteristics. Good representation of both male and female, good representation from all the relevant age groups. So even more than 60, there were about 11% subjects in the study. The mean age was 45. So basically a good representation from in terms of age group and gender, and also the risk of infection. So there was some representation from people who are at higher risk of infection, which includes the because of the profession or age or comorbidities. And there were people at medium, and there were people there is general population or average risk of infection. So overall, the population was well represented. This is the antibody response that was seen. So here you can see yeah, so this is the receptor binding domain RBD specific IgG antibody response. So overall, this is a placebo group, almost no response, and people who were vaccinated, they had a very good response to the vaccine. And this response was seen to all the age groups. This is very important that even above the age of 60 years, there was a very good antibody response. And similar findings we'll see in terms of neutralizing antibody. There was a very high response in patients who were vaccinated, people who were vaccinated, and again, even in the age group above 60 years, there was a good response. So this data also clearly suggests that this vaccine will work well in all age groups and between males and females, no difference. So a very effective vaccine. So this is actual data in terms of efficacy. So here we can see on in 21 days after first dose, 16 subjects in the vaccine group out of almost 15,000 developed were positive or had COVID-19 infection. And in the placebo, 62 out of around 5,000. So 1.3% patient in placebo group versus 0.1% in vaccine group. They developed COVID-19 in the first 21 days. This gives an efficacy of around 91 to 92%. And there were no major differences between gender, and even in terms of age, or the age of 60 years, the efficacy was found to be more than 90%. So here is the results after dose 2. So 28 days after the dose 1. So just when the dose 2 was given after that. So here again the efficacy was good. So overall, so sorry, this is after the first dose, and this is after second dose. So 73 to 87% efficacy after the first dose. At 14 days, 87% efficacy, 91% after the second dose. So that suggests that even after the first dose, there is good efficacy, and the second dose only increases the efficacy. So this data clearly shows that this vaccine has a very good efficacy. I'm not showing the safety results, but because there were no major safety findings in this study. But overall, this vaccine was found to be safe and efficacious. Now the question is, this data is up to 28 days. The study went on up to 6 months, and they haven't published that data. So we don't know what's the efficacy over a period of time. So we know that over a period of time, the antibody titers may come down, and whether that will increase the risk of infection or not, we don't know. But that's a possibility. And that's probably why in different studies, we get different efficacy rates. If you study for a shorter period of 1 month, you may get better efficacy, and over a period of 3 months or 6 months, you may get little lesser efficacy. And that's not the only reason, that's one reason. But when we design a study, for example, in this study, the COVID-19 was detected when subjects had some symptoms after vaccination. And when they had some symptoms, they had to talk to the investigators, and then investigator would talk to the sponsor, and based on some assessment, a decision was taken to do the RT-PCR test, and then the test was done, and it was reported. So, but different studies may have different design of detecting COVID positive patients. So because of these differences in studies and the study design, the duration over which it is studied, there will be differences in efficacy rates reported by different vaccines. But because these vaccines target the same protein, and because the development in formulation has advanced so much that there won't be any difference because of formulation, and even the strategies that people have selected to develop the vaccine, all of them are valid strategies. So from all that, it is not expected that there will be a difference between two vaccines in terms of efficacy and safety. And if we really want to say that some vaccine is better than the other, then they have to be compared in the same trial in a large number of patients. And that has not happened, and no one is going to do that. And so that's why we see some differences in efficacy rates, but overall, what is expected is that all vaccines will have almost similar efficacy and safety. So this is the same data in terms of Kaplan-Meier curve. Here, those who are not vaccinated are likely to have very high risk of infection compared to those who are vaccinated. They will not have such a high risk of infection. And we can see over a period of 80 days, the change in the risk is very small. So this is another evidence that the protection may last very long than we think. So how long will the protection last? That's also a question, because we have been through 6 months or less than a year's time after the vaccines are approved. So we don't have very long-term data to say whether how long any vaccine is going to work. So it will come over a period of time. But if we look at the antibody titers of patients who have been infected uh, with the COVID-19 virus, you can see even after the 4 months, there are significant levels of antibody titers. And this is just an antibody. We also know that there will be a cellular response, which is equally or more important. We don't know how long that stays. So these responses, we expect that they will stay for a very long time. Other important part is there are also, we know that once there is an exposure to an antigen, in terms of immune response, there are development of memory cells. And these memory cells, they get activated when there is a repeat infection or a repeat exposure to the antigen. And that is the most important driver of response in a repeat infection. So that information also we don't have, but what we know from the way immunology works is that there are going to be memory cells after vaccination. And even after many months, maybe even after years, or maybe even lifelong, there will be a better immune response to the virus than without vaccination. So that's what is expected. The challenges with the influenza virus has been that the virus keeps mutating very fast. So the speed of development of vaccine for influenza compared to the speed of mutation, there is a difference. And that's the reason there have been challenges in bringing influenza vaccines or very effective influenza vaccines. But with COVID-19 vaccine, within a year's time, so many vaccines have come, or they are still coming. And now the systems are so well established that even when there is a mutation, in a very short time, the vaccines can be modified to take care of that mutation. So that's why it's expected that the vaccine response to the current epidemic is going to be unprecedented, is going to be one of the greatest that we have seen in the human history. So that these are the expectations, but we should also know what are the limitations. So there has been a long-standing challenge with vaccine development about what is the right dose. And why the dose is important in vaccines is beyond a particular dose. If we give a larger dose than that, then immune response are actually worse. And there is always a safety concern with that. So have we selected the right dose? And with such a faster development of vaccine, we don't know the true answer to that question. So that is a question that still remains. Then there is another question of site of action. So we know that COVID-19 majorly is a respiratory infection, But the vaccines that have been developed largely, they have been systemic vaccines to be given intramuscular or intravenous. And now there are some vaccines that are coming targeting respiratory tract. So these differences, what is going to be the impact of that? So we don't know. One thing that is postulated is that even if the current vaccines are systemic, they will protect against systemic infection, which is viremia, which is the most important cause of hospital admission, ICU deaths, or acute respiratory syndrome and death. So that will be prevented, and that's actually the major goal of developing these vaccines. So that will be achieved even with a systemic vaccine, and there will always be some protection at the respiratory level also. But obviously, a respiratory vaccine is an attractive option. Then the immune response, we know that the virus brings about Th1 response, and there has been a debate that vaccines shift the immunity more towards Th2. And there has been a long-standing debate that because of this, there is, since the vaccination started a couple of decades back, there has been an increase in Th2 diseases like asthma or allergy diseases, allergic rhinitis, atopic diseases, or even the autoimmune diseases. So this debate has been there. We don't know an answer to that. But these are the things we have to keep in mind, that mass vaccinations may have some of the possibilities that we are not currently aware of. So also duration of protection, we still don't know how long these vaccines are going to protect. Is that duration going to be enough for us to uh, to completely eradicate this virus? So we still don't know, but based on how immunology work, it appears that the duration of protection will be enough. And then the long-term consequences also, we don't know of any of the vaccines. So vaccines are single dose or 2 doses. They are given in very small quantity. So it is expected that there will be no long-term consequences of vaccines. This is different than from the drugs which are given every day over a very long period of time. So that's why the expectations is that there will be no long-term consequences. But then there are no studies to make that claim that there are no long-term consequences of vaccination in general. And the long-term consequences may actually be positive. So the possibility of being positive is as high as long-term negative consequence. For example, herd immunity or virus eradication. We may develop immunity against COVID-19 virus at a herd level, and it's feasible, or it's actually theoretically possible that this virus may get completely eradicated. So, or this virus may get mutated into a version which is less infective and less dangerous. So all these positive possibilities are there. Then negative consequences, we talked about autoimmunity or immune reorientation. Our immunity shifts from Th1 to Th2. That is a possibility we don't know. Also, the natural response to react to a virus is to have the intensity of reaction maximum at the site of infection, that is in the respiratory tract, while the vaccines are systemic. So will the system change from local to more systemic level? So even that we are not aware. So and evolution, whether virus infections are a form of evolution. So whether because of this environmental changes, air pollution, all these effects, are these viral infections actually evolving human beings or human respiratory tract to deal with these environmental changes? So, and how vaccination will affect that? So even there are many questions for which we don't have an answers, and it will take many decades, years, centuries to get these answers. But right now, what we know is the vaccines that are being developed for COVID-19 are clearly efficacious and safe. There is no data available to claim that one vaccine is better than the other. So we need to understand that the way these vaccines are developed, they are likely to be similar to each other in most of the respects. So So there is no need to take any extra effort to have one particular company's vaccine over some other vaccine. And the last point has been currently a matter of debate, and it may become a bigger debate later on, is whether immunization should be compulsory or should be voluntary. And largely, what has been followed is immunization has always been voluntary. And even with voluntary immunizations, we have eradicated so many diseases. There is no need to force vaccination on people. We need to motivate, but we should not force vaccination. So with polio, we had this experience that polio lingered over for a very long time, especially in Uttar Pradesh, because of people not willing to get vaccinated. But even then, force was not applied. It's still by voluntary in rest of India, we eradicated polio. And the pockets where the polio was there, uh, just by voluntary involvement, education, motivation, it was possible to eradicate. So that's largely currently the debate, and the immunization should basically be voluntary. So that's all from my side. If there are any questions, I'm happy to take.
Okay. Thank you, Rahul sir, for your brief session about efficacy and safety of COVID-19 vaccines. There are some questions from delegates. Which vaccine we should take first, Covishield or Covaxin, and whether we should be afraid of long-term effects of these vaccines?
Yeah. So those are very relevant questions, and the whole idea of my presentation is we should not think about which vaccine we should take. It does not make any difference whether we take Covaxin or Covishield or any other vaccine. All these vaccines, they more or less have similar efficacy and safety, even though the claims may be different because of the different design of the studies they had and different populations that they took. But it's very unlikely that there will be any difference in efficacy or safety of any of these vaccines. So we should not have a specific choice about which vaccine we should take, whether it's Covishield or Covaxin. Whatever is available and whatever we can have, we should take it. Only thing is, whatever the doses that are prescribed. So if there are 2 doses required, we must take both the doses at the prescribed intervals. That's the only thing. And the other question of the long-term consequences, I don't think we should worry at all, because there is no evidence to show that there are any long-term consequences. These are the hypotheses and theories that are being evaluated, and we never know. There may be positive consequences. There may be very good positive long-term consequences. So at this moment, deciding anything, thinking about long-term is not going to help. So we should not be worried, but it's a good area of research. If someone is interested, it's a good area of research, especially COVID-19 vaccination offers a unique opportunity where a large population is vaccinated at the same time. And this is the right time to study these people over a period of time to see if there are any long-term consequences. But we should never be worried about that.
Second question is that can asthmatic or any other respiratory disorder patient can take vaccine and whether they will face any problem?
So asthmatic patients, these are the patients who asthma or COPD or any respiratory disease. These are the patients who are in a larger need of vaccine, because if they have infection and they have simultaneous asthma attack, it will be very difficult for them. So mortality has been high in these patients. So that's why these are the patients who should first take the vaccines. It's very important for them to get vaccinated, and vaccines will be effective in them. And whether it will have any adverse effect on asthma, there is no reason to think that, because we have such good treatment for asthma now that any asthma can be controlled. So there is no reason to think about any adverse effect of vaccine on asthma. So this patient must take vaccine.
What happens when we are unable to take second dose of vaccine within a specific duration mentioned by the guidelines?
Yeah. So this duration is not a very hard and fast duration. It's not that if it says 12 weeks and if you take in 13th week that will not have an effect. That's not the case. we should take it within the duration, and if there is a delay, we should try this to take the second dose as early as possible. But if there is a significant delay, for example, instead of say the prescribed duration is up to 12 weeks, and up to 24 weeks we don't take the second dose, then in my view, I think the best way is to repeat the complete immunization. Start with the first dose again.
Is a change in the dosage. Like in Johnson vaccine, there is only one single dose, whereas Covishield has two doses. Why such things are there?
Yeah, so it depends on a lot of factors. It depends on uh, what how that vaccine is made. So we talked about strategies. So what is the strategy, and what excipients, what other agents are used in formulation, and what dose of the vaccine has been selected. So all these factors determine whether there is a need for a second dose or not. So many vaccines, for example, many vaccines will require a second dose because maybe the first dose is not enough to reach a level that will sustain over the period of time we want, and the second dose will give a booster to that, and then the antibody response will be more sustainable. But some vaccines, the first dose quantity itself is enough to give that response. Some of the vaccines, like this Sputnik V vaccine, the first dose of vaccine is actually enough for to give the immunity. But they have this second additional dose. So there, if we miss the second dose, it's not going to make a bigger difference. But because it's been prescribed, we should try and take the second dose.
Why are we are not using other antivirals than remdesivir?
So there are many antivirals that have been researched, and it does not make more difference in terms of efficacy of one antiviral over the other. The advantage of remdesivir is it is intravenous, and why that is important is because the blood levels are reached very fast, and when there is a viremia, when there is virus in the blood, which is a major cause of deaths or ICU admissions or complications of COVID-19, those can be prevented, and that's the main reason why remdesivir is used so commonly, especially in the hospital setups in the ICUs. But there are other antivirals like favipiravir. It's the most commonly used antiviral in India. So there are antivirals that have been used, and they have been shown to be efficacious. Important thing to know about antivirals is especially when like favipiravir, they are going to be effective only when they are given at the beginning of the infection. That is within first week of infection, or within first 5 or 3 days when the symptoms start. Because once the viral load is reached its peak, then after that, reducing the viral load has not been shown to change the outcome. It's only before the viral load reaches the peak and you give an antiviral uh, and you reduce the peak. So only that has been shown to be efficacious. So all antivirals given before uh, uh, within first 5-7 days of infection, they have good, very good efficacy.
So looking at the development of these new variants like Delta in COVID-19, what will be the future prospects or aspects of vaccine development?
So for vaccine development, the story will be similar to what happens with influenza. There will be a change required in the vaccine to have efficacy against the new variants that are coming. So these things, so all vaccine manufacturers, they will keep changing their vaccine to have efficacy against the current variants. But till that time, the existing vaccine, what efficacy does the existing vaccine have, is also very important. And as we saw, that all these vaccines are targeting the receptor binding domain of spike protein. So if the current variant, the mutation is not in that part of the virus, it will not make any major impact. So vaccine efficacy and safety will still be there. The only challenge comes when that there is a mutation in the receptor binding domain, and that will need a new vaccine with additional antigens in it.
One more last question for you, sir. In post-COVID, steroidal drugs are used for life-saving, but for diabetic patients, it may be harmful. So, what alternative treatments for diabetic patients suffering from the post-COVID can be used?
Sorry, I did not get the first part. What did you say?
In post-COVID, after COVID infection, steroidal drugs are used for life-saving purpose, and it may be
Steroid.
harmful for diabetic. Yeah, and it may be harmful for the diabetic patients. So, is there any alternative treatment for diabetic patients post-COVID?
Yeah, so one important thing here is the post-COVID disease. Again, it's a new thing. A lot of research is already happening, but currently, steroids appear to be the most important drug to prevent long-term consequences in the post-COVID time. So it is an important drug, and it should not be stopped or reduced because of diabetes. So it should be given the same way it's given to non-diabetics. There should be no change. What are the effects on diabetes? So the effect on diabetes obviously is the increase in blood sugar level, and that can anyways be very easily managed with so many medicines currently available, or if required with insulin. And these steroids are required only for a short period of time. So but it's very important that they should be given because currently they are the most effective drugs in that duration.
Okay. Thank you, Rahul Kodgu, sir, for your engaging talk. And on behalf of Dayanand Education Society, Dayanand College of Pharmacy and Dayanand Institute of Pharmacy, I'm thankful to you for such a wonderful session. And now for next session, I will hand over the session to Samip Sononi, sir, our program director. Samip Sononi, sir.
Thank you very much.
Thank you, sir. Thank you, sir. A warm good afternoon, one and all. Myself, Sonone Samip, coordinator of this e-conference entitled as COVID-19 Era, Need and Challenges in Healthcare and Pharma Sector. I take this opportunity to share my view. As we know, from last 1.5 years, we are faced lots of difficulties due to the COVID-19. To overcome this problem, we try so much things. Lots of speculation is going on. So, to overcome this thought, we think or we take an expert assistance. That is one of the objective of this e-conference. To enhance the understanding of COVID-19 deterrence method in an effective manner and provide an outlook on the challenges brought by the COVID-19 pandemic, to explore the opportunity and prospect for discovery and resilience vaccine development. That are the three different objectives of this e-conference. To get an answer of this in effective manner, these are the different outcomes of this e-conference. I am very much thankful to Dr. Dinesh Dhumal sir, Dr. Jagdish Mahale sir, Dr. Prajakta Kulkarni madam, Dr. Debashish Naik sir, and Rahul Kodgu sir for accepting this invitation within a short period of time and make this conference a successful. This conference clear our doubt related to the safety of vaccine and also gives idea regarding different thoughts working behind the vaccine development. This gives information about the various animal model used and different tests used for the identification of coronavirus. We are very grateful for such a huge response for the conference for all over the globe. This motivates us to conduct the conference seminar on different topic in nearby future. Thank you once again. Now I hand over the mic to Ms. Rajmani madam to give a vote of thanks.
Thank you, sir. Now we are heading towards end of this conference. It's an honor to offer a vote of thanks for such a grand international e-conference. On behalf of my college and entire management team, I would like to thank our chief guest, Dr. Jogendra Singh Ji Bisen, Pro Vice Chancellor Swami Ramananda Tirth Marathwada University, Nanded, who gave his vital time from his busy schedule and inaugurated the conference with an enchanting and memorable poem. I also express my gratitude towards guest of honor, Dr. Suresh Ji Bhattad, Management Representative, Dayanand Education Society, Latur, who honored this conference with his inspirational message. His able guidance has always encouraged us. I take this opportunity to exchange our most sincere thanks to the enthusiastic and motivating management team of Dayanand Education Society, Latur. President, Honorable Shri Lakshmi Ramanji Lahoti, Vice President, Honorable Shri Arvindji Sonavne, Vice President, Honorable Shri Lalit Bhai Shah, Vice President, Honorable Shri Ramesh Kumar Ji Rathi, Secretary, Honorable Shri Ramesh Ji Biani, Joint Secretary, Honorable Shri Suresh Ji Jain, and Treasurer, Honorable Shri Sanjay Ji Bora Sir. I thank the entire management team for their kindness, their interest, motivation, and continuous support. If every college has team leaders like you all, I'm sure there is nothing to stop them from achieving success. Dayanand Education Society is blessed with such a great leadership. A special mention to our venerable principal and convener of this conference, Honorable Dr. Kranti Satpute ma'am and Dr. Srinivas Bumrella sir, being the catalyst that inspired us to do our best and stand as a pillar of power. Thank you very much, ma'am and sir. I also express my sincere thanks towards coordinator of this international e-conference, respected Sameep Sonavne sir, Head of Pharmaceutical Chemistry Department, Dayanand College of Pharmacy, Latur. I am also thankful to all the co-coordinators of this conference, respected Yogesh Kulkarni sir, Gopal Lohia sir, Shraddha Tiwari ma'am, and Prakash Shivnechari sir for their constant support. I must thank Dr. Vajay Chaus, Head of Pharmaceutics Department, Dayanand College of Pharmacy, Latur, for his endless efforts. I am also grateful to all the eminent speakers from India and abroad who added their valuable insights on COVID-19. I am fortunate to thank the dedicated and well-motivated team of our college for putting in their efforts to make this conference a grand success. I should not forget to thank all the anchors for their appreciating way of conducting this conference. I would also like to appreciate the enthusiastic participation of all the learned delegates, respected teachers, special invitees, our guests, and your students. We got a huge response from the participants, which will keep encouraging us to organize such conferences frequently. I must thank our technical support team, VM EduLife, without whom smooth conduct of such a great conference was not possible. Last but not the least, a special thanks to all non-teaching and supporting staff who have worked hard to ensure that the program becomes a memorable one. Thank you one and all for making this event a successful one. I appeal all the participants to fill the feedback form. Madam, may I conclude?
Yes, madam, yes, you can.
With the permission of chair, I would like to declare that the session is concluded. Stay happy, stay safe. Thank you all. Do not forget to fill the feedback. It is valuable for us. Thank you.